Hey guys, how is everyone doing? Wow, 282 of you in here. How's everyone doing? How was English? How's English lit? I'm seeing some familiar names in the chat. RS tomorrow. Not bad. Hello. Did anyone print the worksheet? Yeah, it'll be useful if you printed out the workbook, guys. That's what we're going to be doing today. Hi guys, hi Frozen. Familiar names here. Some of you from the boot camp, some of you from our FRL classes. It's good to see you all. Guys, it's bio paper 1 to first science paper out of 6. How are you guys feeling for it? You want a 999? Well, hopefully I can help you. Today, the plan is, guys, we're going to go through the key topics in bio paper 1. We're going to cover some content and we're going to try exam questions as well. We're going to do a bit of everything. So obviously I won't be able to go through every single topic throughout the whole spec. If you do want to cover some topics that I don't go through, we've sent you guys an email. In that email, we've given you cheat sheets. We've given you a bootcamp recording, which goes through every single topic. So you can go ahead and watch that as well. And we've given you this workbook as well. So this workbook will be useful to have. This is what I'm going to be going through today. Well, so many people in the chat. I don't know how I'm going to keep up with everything. I'll try and answer questions if I can. But yeah, I hope you guys will find this useful. We're doing this 6 times, guys. So all the paper ones are free. So you can see here, we've got chemistry this Sunday. So 2 times in a week. Then we got a break for half term. First day back after half term on the 1st of June, we'll be having Nav teaching you guys physics for the paper 1s. And then we've got the paper 2s on the 7th of June, 11th of June, and 14th of June. And then, guys, you're done. That's it. You can enjoy your summer holidays, unless some of you got those extra exams at the end of that. So, guys, try and absorb as much as I say today. It's going to be a lot. If you want to participate in the chat, remember to subscribe as well. I think that's the only way you can, that's the only way you can chat here. If you guys do have any questions about like things not being sent to you, I know some of you guys signed up and then I think you typed in the wrong email address, so we won't send you guys the workbooks and things. So if you do have an issue like that, email help@flashrevisionlab.com. Let me write it down here so you guys can do that. help@flashrevisionlab.com. If the stream goes down, hopefully it does. If it does, bear with us. Sometimes it does lag or it goes blurry for a bit. Usually it corrects itself after a while. So just bear with me. You should be able to hear me. If it does really go bad, then I'll go off and come back on again. And if we need to switch streams, which we never have had to do in the past, we'll email you guys a new link or something like that. All right, cool. Let's go, guys. We are going to go through this workbook, which we sent you guys. So this is the Cram Class workbook. I'm going to switch between this screen with all the workbook content and the cheat sheets. So both of these were sent to you. So I'm going to use the cheat sheets to teach you guys the topics, the content, and then if I want to do some exam questions, then I'm going to go to this screen. So yeah. Let's get started then, guys. So key topics for bio paper 1. We're going to start off with topic 1. I'm going to try and give you guys some breaks as well. So for bio paper 1, there's 4 topics. So after every topic, I'll try and give you guys a break, maybe like a 10-minute break. But I'll ask you guys how you're doing, whether you want a break or not, and you guys can let me know. Okay, let's go. If the quality is bad, guys, I apologize. Hopefully it will improve after a bit. So what we're going to do, I'm going to go through the first few key topics in bio paper 1. So first of all, we have eukaryotes and prokaryotes. Okay. In prokaryotes and eukaryotes, first thing you need to know is the differences between prokaryotes and eukaryotes. Okay, so this is a memory trick I like to use to help people remember the differences. So eukaryote, you sounds like you, as in you, an animal, a human. Animal cells are eukaryotic cells. So in an animal cell, you have a nucleus. So eukaryotes have a nucleus. Nucleus has the letters EU in it. Ooh, there we go. That's how you remember eukaryotes have a nucleus. Prokaryotes, pro sounds like no, they don't have a nucleus. So no, they don't have a nucleus. Examples of eukaryotes are you. So these are animals, animal cells, as well as plant cells. And prokaryotes, the opposites, we have bacteria cells. So that's the difference. Let's look at this. In the cheat sheet, this is what we say. So eukaryotic cells have a nucleus. Prokaryotic cells do not have a nucleus. Eukaryotic cells are larger and more complex, whereas prokaryotic cells are smaller and simpler. Examples, animal and plant cells, and examples of prokaryotic cells are bacteria cells. So 3 different types of cells you guys need to know about. Animal cells, plant cells, and bacteria cells. Okay, can anyone tell me what's the difference between an animal and a plant cell? What do plant cells have that animal cells do not have? That gets asked quite a lot here. What do plant cells have that animal cells do not? Let me know in the chat and we'll add it here. Plant cells have, I'm seeing the answers here. There we go. Chloroplasts, permanent vacuole, cell wall, as well as everything else an animal cell has. So only plant cells have a chloroplast, a permanent vacuole, and a cell wall. Okay, so let's talk about what each of these things do. Let's go back to our cheat sheet here. So a cell membrane is the outer part of an animal cell. It controls what goes in and out of a cell. Cytoplasm is like a liquid, it's like a gel, and that's where all the chemical reactions occur. An example of a chemical reaction that occurs here, we got anaerobic respiration happens here. We've got the nucleus. The nucleus contains genetic material, so DNA, and ribosomes is where protein synthesis occurs. So that's where proteins are made. Amino acids combine together to make proteins. And then we've got the mitochondria. This is where aerobic respiration occurs. We're going to talk about aerobic and anaerobic respiration again in topic 4. It's one of the most important topics, guys. Now, for the plant cells, we got a cell wall. What do you need to know about the cell wall? Cell wall is made out of cellulose. So glucose gets converted into cellulose. And what that does is it provides support to the cell. It makes it stronger. So whenever you get a diagram of an animal cell, guys, or a plant cell, for example, the inner one is the cell membrane, the inner wall. The outer wall is the cell wall. Okay, so make sure you differentiate the difference between them. We've also got the chloroplast. The chloroplasts have chlorophyll in it. The chlorophyll is a green pigment, and that's where sunlight gets absorbed for photosynthesis. Yes, I'm seeing the correct answers here. All right, so that's animal and plant cells. Then we've got bacteria cells. So bacteria cells are a bit weird. They've got, yes, they do have a cell wall. They've got a cell membrane. All cells have a cell membrane. It's the thing that's holding the cell together. They also got cytoplasm. They've got ribosomes. They don't have mitochondria. Why do bacteria cells not have a mitochondria in them? Let's make a note of that, actually. No mitochondria. How is it a bacteria cell can't have a mitochondria? Does anyone know the answer to that? So remember, the mitochondria is where energy is released. It's too small. Yes, well done. They're smaller than a mitochondria. Bacteria cells are so small that they're smaller than a mitochondria, so they don't have any. But they do have ribosomes. The main thing, remember, bacteria cells are prokaryotic cells, so they have no nucleus. Instead of a nucleus, they have a strand of DNA which floats around in the cytoplasm, and they also have plasmids. Plasmids are these like rings, and these are like rings of DNA, which are also floating around in the. All right, so that covers eukaryotes and prokaryotes. So these are like the key topics that we think are the most common ones that come up, and you can see how common they come up here on these tables, by the way. Eukaryotes, prokaryotes, animal cells, and plant cells comes up like almost every year. So make sure you know all those definitions. Make sure you know the differences between plant cells, animal cells as well as the differences between prokaryotes and eukaryotes. So we got those as very common topics. Next, what we've got after that is microscopes. Guys, this is our first required practicals. Required practicals are extremely important because they have to come up. So there's like a certain number of required practicals for every paper. They have to ask you questions on those. So they are guaranteed to come up. Which one comes up, we don't know. So it could be microscopes. It could be osmosis. It could be enzymes. It could be food tests. But they have to come up. So those are like really important for you guys to go through. So what do you need to know for microscopes? Most questions are these things on the left-hand side. The difference between light microscopes and electron microscopes, as well as a magnification equation calculation. You don't get that many calculations in biology compared to like physics and chemistry, but when they do, it is very often a magnification calculation. Okay, so those are one of the ones that we're going to practice in a bit anyway. So first of all, what's the difference between light microscope and electron microscope? Put that in the chat right now if you guys know. Difference between electron microscope and light microscopes. So this one is a required practical. Let's see what the chat is saying. Light microscope, higher, no, not higher magnification for light microscope. Electron microscope has a higher magnification, a higher resolution. So it can zoom in more. It can zoom in more to images. It has a clearer image as well. Electron is better. It's just more expensive. It's like much more expensive. And you've probably never seen one because only scientists can use them. They're very complex to use. So yeah, that's what I'm seeing here. I'm getting the correct answers here in the chat. So this is what we've got here. Light microscope obviously uses light to make an image. It doesn't just use light. It can use a mirror as well, where it reflects light from the surroundings to illuminate the sample that you're looking at. Electron microscopes use electrons, more magnification, more resolution. But electron microscopes are more expensive. They're very complex to use. Good thing about them is you can see smaller structures, whereas for light microscopes, if you were to look at a cell in a light microscope, this is what it would look like. So you'd see the cell membrane and you'll see a nucleus. That's all you'll see in a light microscope. So if they showed you a diagram which only showed you a cell and a nucleus within it with nothing else, and they asked you what type of microscope, has this image been taken from? You'd say a light microscope. But if they give you one where you can clearly see all the other things, so ribosomes, mitochondria, they're like usually like oval shapes. Ribosomes are like little dots. Mitochondria are like ovals. Then you'd say that that comes from an electron microscope. Yes. Okay, cool. Next. This calculation, magnification. What's a good way to remember, to memorize the magnification equation, guys? Because remember, you don't get an equation for biology. What is the memory trick to remember the magnification equation? I am. Yes, I am is the way to remember it, guys, if you want to memorize that. Because they don't always give you the equation in the questions. So I am. So the I is for image size over actual size times magnification. Okay? Remember that. Write that on your paper if you want to remember. Like right at the start of the exam, just write it on like a corner of a page. Image size is, magnification is equal to image size over actual size, and you can use that triangle to like rearrange as well. That calculation comes up a lot, guys, a lot. Okay. With that, there's also a conversion that you guys need to do. You need to be able to convert from micrometers to millimeters. This is also something that comes up a lot. How do you convert from micrometers to millimeters, guys? Let me know in the chat. What do you guys think? Micrometers to millimeters times by 1,000. Actually divide by 1,000, guys. Micrometers to millimeters, you divide by 1,000. Millimeters to micrometers, you times by 1,000. People always make that mistake. Okay, here we go. To go from micrometers to millimeters, divide by 1,000. Millimeters back to micrometers, you times by 1,000. So remember that, guys. Okay. So those are the main things that they ask. Occasionally, they ask you about how to carry out an how to view something under a microscope. If they do, then these are the steps. First thing, you get a slide. On the slide, you add a drop of water. Then you add the sample that you're trying to view. So usually it's like an onion, a layer of an onion epidermal tissue. So you peel that off the onion, you put it on the drop of water. Then you add iodine because that is like a stain. If you look at an onion layer in a microscope as it is, you wouldn't be able to see anything because it's transparent, translucent. So you need to stain it so you can see all the structures. So you add iodine there. Then you have to add the cover slip. I've seen this question come up before. You have to add it at like an angle carefully to ensure there's no air bubbles. Because remember, you have a layer of water, you're putting another glass sheet on top of it. You don't want any air bubbles in it. Otherwise, that's going to affect your image. You're not going to see any of the cells clearly. So you have to add it carefully. And then you always start with the lowest objective. Two types of lenses here. We've got the objective lens here. So this is the one which has 3 different magnifications. So this might be like, I don't know, times 10. This one might be times 20. And this one might be times 30. And then you have the eyepiece lens at the top. The eyepiece lens cannot change magnification. It only has 1 magnification. So that could be like, I don't know, times 20. So that magnifies something 20 times. What you have to do is you need to multiply that with the objective lens. So if you're using times 20 eyepiece lens and you're looking through the times 10 objective lens, times 20 times 10 is equal to times 200. You multiply them together to find the overall magnification. So you can switch through these. So you start off with the lowest objective length. So you'll start off with the times 10 first, and then you'll try and adjust the knobs to focus on it. And then if you want to zoom in even more, you'd switch the objective lens after that. Okay, so let's go back to our topics that we have. Microscopes. Those are the key things that you need to know about microscopes. Next, diffusion, osmosis. And I am going to include active transport in here as well, because I always teach these three things at the same time. Because once you know, guys, it's really easy to know all three of them. Okay, so this is all to do with the movement of particles. Okay, the differences between them is where are the particles moving from and where are they moving to. So the definition always sounds very similar. So look what I've done here. Let's go down here. I've written a rough definition here. It says that net movement of something particles from an area of something concentration to an area of something concentration. What do you guys think these missing words are for diffusion? And what I'm going to do to show you guys that they're pretty much all this, like they're pretty much very similar to each other, I'm going to copy and paste this definition, and we're just going to literally tweak a few words. If you know one, it's very easy to know all three of them. What are we going to say? High to low. There we go. So diffusion, the net movement of particles from an area. So here, there's no word. For this one, there's no word. So the net movement of particles from an area of high concentration to an area of low concentration. Okay, so that's the basic one. Diffusion is the basic one. Osmosis. Osmosis. Think of it as diffusion with water. Okay, so it's the same definition as water. It's the same definition as diffusion, but we're going to add the word water in the definition. So we're going to say the net movement of water particles from an area of high water concentration to an area of low water concentration. We're just adding the word water here, but there's something extra we need to say. What is that extra thing we need to say if we're defining osmosis? And you can use the M in osmosis to remind you of that. Can anyone let me know? I'm going to write it M as like a capital here to remind you guys. You need to talk about a partially permeable membrane. Okay, good. So I'm going to write that down as well. The M you can use to remind you of the word membrane. It's through a partially or semi-permeable membrane. Okay, now you got to be careful here with this definition because you might have been taught it differently in school, and that's fine. You can also say, okay, this is the way I tell all my students to remember it because it's similar to diffusion, yeah? But you might have learned it like this. The net movement of water particles from a dilute solution to a concentrated solution. That's fine as well, across a partially permeable membrane. You can say that as well. That is correct as well, okay? What you can't say is the net movement of water particles from an area of high concentration, and miss out water here, to low concentration. That's wrong. You can't say that, okay? So learn it whichever way you want, but I always tell my students to learn it this way because if you know diffusion, you know osmosis, you know what to add. Okay. Next one, active transport. If you want to do active transport, if you want to memorize active transport, then you just need to say that active transport is the opposite opposite of diffusion. Okay, so what's the difference? The net movement of particles from an area of, instead of high to low, you're going to say low to high concentration. Exactly. And the extra thing you need to say here is that it requires energy. Where does that energy come from, guys? Let me know in the chat. Which requires energy from, there we go, people are saying it, from respiration. All right, so diffusion, let's add a few more things here. Diffusion goes down a concentration gradient. So that means it goes from high to low. Active transport goes against the concentration gradient. There we go. People are saying it in the chat. Well done, guys. And there we go. That's it. That's it. Okay. That's the definitions of each one. That's the difference in the definition of each one. What else do you need to know? So for diffusion, let's jump to diffusion here. Diffusion, you need to know examples of diffusion. How to increase the rate of diffusion. You increase the concentration gradient. So that's the difference between the area of high concentration to the area of low concentration. Increase the temperature. That causes the particles to move around faster. So the particles can move from the high to low concentration much faster. And also increasing the surface area to volume ratio. The smaller an organism is, the greater its surface area to volume ratio. Examples of diffusion. We've got the alveoli in the lungs. We've got leaves in plant. So the alveoli in the lungs are for a gas exchange. These are all the adaptations that it has. This is why it's so good at taking in oxygen and adding it to the blood so that it can be circulated around the blood. Leaves in plants, they're adapted for gas exchange as well. They need to take in carbon dioxide for photosynthesis. They need to give out oxygen as well. These are the adaptations for those. We've got in the small intestine, so we're going to talk about the digestive system in the second part of this live. We've got the villi. So the villi are just these like tiny projections that stick out in the small intestine, and they increase the surface area even more. And here are more of the adaptations that help it. And finally, the one that gets asked sometimes, not as much, you got gills in fish as well. Again, that's how fish breathe. They need to take in oxygen from the water. Look at the adaptations here. So if you look at the adaptations, guys, it's always similar. Large surface area. They have thin walls or short distance for diffusion. Some of them have a large blood supply as well. So like gills in fish have a large blood supply. It makes diffusion more effective. Same with the alveoli in the lungs. Leaves don't have that because leaves don't have blood. Villi also have a large blood supply as well. So if they ask you about any four of these and they ask you how are they adapted, you can talk about they have a good blood supply, large surface area as well, and thin walls as well. Okay, so diffusion. Osmosis. Out of the three, osmosis is probably the most important because there's a required practical on it. So we know the for it now, okay? Because you know diffusion, you know that osmosis is diffusion with water, right? So an example of osmosis you need to know is in a root hair cell, how they take in water. They have a high concentration of water in the soil, high water concentration. They have a low water concentration in the root hair cell. So, like the definition, the water goes from the high concentration to the low concentration. That's how it takes in the water. And it goes through the partially permeable membrane, which is the cell membrane and the cell wall. Okay, so the required practical, how does this work? This comes up a lot, guys. Required practical. This is the one with the potato. Yeah, potato cylinder. I've seen a carrot, I think, in the past. Have you guys seen that in a past paper? Anything else you guys seen? It's usually some sort of plant tissue. If you look at the spec, it says plant tissue. So putting some sort of plant tissue in solutions of sugar, usually. Okay. Yeah. So what you need to do here, guys, you need to get potato cylinders. You need to cut them into equal size and you need to immerse them into sugar solutions. There's sugar solutions of loads of different concentrations. Okay, so you might have one which is pure water. You might have one which is a very low concentration. You might have one which is a very high concentration. Usually, the concentrations will look like this. We're going to see this, and you'll see this in some of the questions. 0 moles per dm cubed, 0.1 moles per dm cubed, and so on. It might go all the way up to, I don't know, 0.6 moles per dm cubed. These are just concentrations. So this is pure water. This is a very weak solution. And this is, this is a very concentrated solution. So what you do is you put these potato cylinders or plant tissue cylinders into the solution and you leave them for a few hours. Could be 24 hours. I think in one paper, it was like half an hour. And then once you've done that, you take them out. You have to blot them dry with like a paper towel. That's because you need to remove the excess water from them, and then you weigh them again. So you weigh them before and you weigh them after, and then you find the change in mass. Sometimes the mass will increase. If the mass increases, it means water has entered due to osmosis. That's because you have a high water concentration outside, you have a low water concentration inside. Remember, you have to say water concentration. Don't say concentration on its own. You won't get the marks. Or it could get smaller. Sometimes it gets smaller. This is when you have a high water concentration inside the potato, a low water concentration outside. So the water leaks, making the potato shrink. Sometimes the potato doesn't change shape at all. If the potato doesn't change size or shape or mass, that means there is the same water concentration inside the potato and outside. So whatever concentration the solution is at that point, that is the concentration inside the potato. So if the mass does not change, it means the concentration outside and inside are exactly the same. Okay, so control variables, things you need to keep constant: the volume of the solution, the temperature, the time you leave them in for, the type of potato, things like that. Things you need to change are the sugar solutions, the concentrations of those. Things you need to measure is, so that's the dependent variable, that would be the mass. And then a calculation that you'll be asked to do, this comes up a lot, is a percentage change calculation. So you see here, if you started with a potato that was 1.3 grams and then ended up as 1.51 grams, they might ask you, find the percentage change in the mass. What you need to do is this equation. I would tell people to memorize this equation if you guys aren't good with percentages. Change in mass over the initial mass. Change in mass over the initial mass. Okay, so you would get your in mass, so that's your 1.51 minus your 1.3, that's your change in mass. Divide that by your initial mass, so not your final mass, the one that you start off with. Divide that 1.3 times it by 100, and then you'll get your answer, 16.2%. Okay, very common calculation. Right. I think those are the key topics that I wanted to go through for this. Let's try some questions, guys. You guys tell me in the chat. If you guys know the answers to these, fill them in if you've got them in front of you. Let's go. I'll give you guys a break once we hit about, I don't know, let's say 6:30. Or if we get through all of these questions, I don't know if we will. Let's see. So What contains the genetic information, guys? Let's go. Quick. Nucleus. Yes, well done. I just told you guys that at the start of the class. Mitochondria. What does the mitochondria do? Guys, do not say it's the powerhouse of the cell. Please don't say that. You won't get a mark for that. Site of aerobic respiration. I like that. Yes. Site of aerobic respiration. What controls the movement of substances into and out of the cell? Cell membrane. Yes, it is. Well done, guys. All right. What is the function of chloroplast for 1 mark? What is the function of chloroplast? It's 1 mark. You can get away with just literally saying one word here. Saying photosynthesis is enough. You can say they absorb sunlight for respiration. Yes. The answer here is photosynthesis. Remember, the chloroplast is what has the chlorophyll in it. Photosynthesis. Photosynthesis. It has the chlorophyll in it, and that's what absorbs the sunlight. Yeah. You can also get marks for saying producing glucose as well. We'll talk about photosynthesis in the 4th session today. One type of cell that does not contain chloroplast. Those of you who was on my TikTok live yesterday, we went through this this page, actually, the first page. What's an example of a type of cell in a potato plant that doesn't contain chloroplast? Remember, chloroplast and chlorophyll is what makes a plant green. So think about what part of plant is not green. Yeah, the roots. So the root hair cells is one. You can also say xylem. You can also say the xylem cell as well. We'll talk about the xylem later. The xylem is made out of dead cells. Okay, that's in the 2nd session. We'll be going through the xylem and the phloem. Okay, here we go. Required practical question. So let's look at this method for that potato experiment. The student investigated the effect of salt concentration on pieces of potato. They cut 3 potato of the same size. Okay, I like that. Same size. They're keeping that consistent. They recorded the mass of each potato piece. That's the initial mass. And then they added it into 0.4 mole per dm cubed solution of a beaker, and they placed each potato into the beaker. After 30 minutes, they only did it for 30 minutes, interesting. They removed the potato piece. They dried it with a paper towel. That's important. Good. Remember, that's to remove excess water. I've always seen that as like a 1 marker before. And then they recorded the mass of each potato piece after. And then they repeated it at different concentrations of salt solutions. Pretty much what we're expecting here, okay? Independent variable. What's the independent variable? Remember, the independent variable is the thing that you are changing. Dependent is what you're measuring. Concentration of salt solution. Excellent, yeah. This, I would say, is the dependent. That's what you're measuring. This is a control variable. Control variable is what you're keeping the same. This is control variable as well. You're keeping that the same. So independent is what you're changing. Someone came up with a memory trick for this the other day. I forgot what it was. If you're here, can you tell me, please? Independent, and then dependent is what you're measuring. And then the control, what you're keeping the same. Remember that. Something to do with ice cream, yeah. Sid Sam. Sid Sam. Sid Sam. Does that work? Depending. Oh. Control, independent, dependent. Same. Measure. What's the A for? In Sid Sam. Adjust. Okay, I like that. Right, guys, here we go. Control. This is what I like. I like it when we come up with memory tricks on the spot. Control variable, independent, dependent variable. Control is what you're keeping the same. A Adjust. This is what you're adjusting, what you're changing. The independent variable. Dependent variable is what you're measuring. Shall I make a video on this, guys? Let me know. Shall I put a TikTok on this? It's a good one. I like that. Would this work for OCR? Yes, it will work for any. This is just a general thing you need to know for science, so it would work. So control, independent, and dependent. Make sure you know the differences between them. Why did the student dry the surface of each potato cell with a paper towel? Guys, I told you this already. Say it the way I said it. Remove excess water. Because when you're weighing it, guys, you don't want water on it. You're trying to weigh just the potato. If it's wet, then it's got a lot of extra water on it. So you need to remove excess water so that you're not weighing the water as well. Here we go. Calculation. This is exactly the type of calculation I expect in this type of question. They've been nice here and they've given you the equation, yeah? They don't always give you the equation. So please remember how to work out percentage change, guys. So change in mass over initial mass. They've said increase in mass over starting mass. If you guys got a calculator, work it out and let me know what you're getting. We got people getting 8% already. Let's see. So change in mass is 2.5 to 2.7. So it's 2.7 minus 2.5. That's our change over initial mass. Don't do over 2.7. Do it over 2.5. Remember, it's percentage. We're going to times that all by 100. And I think you should be getting 8%. That is absolutely right, guys. Well done. You guys are smashing it. Okay. Next one. The student used the results from each potato piece to calculate the mean change in mass at each concentration. Okay. Complete figure 1. You should label the axis. Use a suitable axis. Okay, guys, I'm going to do this one with you. If you've got it with you, try this out, okay? Because graph calculations will 100% come up in your science exams, yeah? Most likely to come up in a biology paper, a graph question, okay? So 0, 0 goes to 9.8. We need a good scale here, okay? You have to use minimum 50% of the grid, okay? So you can't be doing this. You can't put all your graph here and plot here and end there. If you do that, you're going to lose a mark. So make sure you're using at least 50% of the grid. They're usually nice and they give you like a good grid. So this is going from 0 to 0.4, right? So 0.1, 0.2, 0.3, 0.4. I think that's a good scale to be using. 0.4. Yeah, and we can have a 0.5 there as well. So yeah, this is 2022. It came up in 2022. We've got 0, 0 goes to 9.8. Let's try that, plot that. 9.8. 9.2, 4, 6, 8. 9.8. So we're starting there, 9.8. Then at 0.1, we're going to 9.5. 9.5 is halfway. 9.5. Then 0.2, we're going to 7. 0.2, we're going to 7. Then 0.3, we're going to 0.4. It's quite a sharp drop there. And then at 0.4, we're going to minus 1.4. Minus 1.4, which is around here. It says, draw a line of best fit. What do you guys think this is going to look like? Is it a curved line or a straight line here? You have to judge it yourself. If they say draw a line of best fit and they don't tell you whether it's a curve, sometimes they say a curve of best fit. Sometimes they just say a line and you have to figure out whether you have to do it straight or curve. You have to 0.4. Oh yeah, sorry, my bad. This one's 0.4. Yeah. Sorry. This is 0.4, not 4. So this is a curve, guys, because look, you can kind of see the curve. You need to know the rough shape of these graphs. Whenever it's an osmosis practical, this is how it looks like. It's usually like a sort of curve that gets steeper and then less steep again. So let's try and draw a smooth curve. That was not a good start. My iPad's slippery, so it's quite tricky. Something like that. Okay. How do you know whether to do a curve or a straight line? That's a good question. So some of the graphs you need to know. So like photosynthesis limiting rate graphs, you need to know. So you need to know that's a curve. So if they give you points to plot on that, you need to know that it's going to be curved, and you need to estimate that, right? Otherwise, you have to look at the points, and you have to see if you can make a curve out of this. Here, you can make a curve out of this, right? But imagine if we had loads of points like that, we would need like a straight line of best fit. So you need to judge it. You need to look at the graph itself and see whether you can get a smooth curve. If you can't, or if it's starting to do like weird stuff, if you draw a curve, it starts to do this, then it's probably a straight line of best fit. So you guys need to judge it. Okay. But for this osmosis practical, it is almost always a curved line. Okay. So when they ask you to plot a graph, after that, they do usually ask you a question on the graph. So they will base it off the way that you've drawn your graph. So let's see what the next question is. What concentration of salt solution was equal to the concentration of the solution inside the potato piece? How do you guys think we can work this answer out? Like I said, this is going to be using your own graph. We need to use what we've drawn here. What concentration of salt solution was equal to the concentration of the solution inside the potato? What did I say earlier? It's where it meets the x-axis, Sammy. Yes, exactly. Exactly. So where does this meet the x-axis? What would you guys say the answer is here? That's where it meets the x-axis, right? What is that reading? 0.32. I think that's a good answer to give. 0.32. Okay. This is a common required practical that comes up quite a lot. Okay. Where's the mark scheme for this? We're going to send you guys the mark schemes at the end of this session. We're going to send you guys the mark schemes for all the questions, because I doubt we'll get through all the questions. So you guys can use the rest as practice, last minute practice if you guys want. Explain why the potato piece in the 0.4 mole per dm cube solution decreased in mass for 3 marks. 3 marks means 3 points. It's explain. Look at the command word. Explain. That means you need to say why it's doing it. Okay. If they say describe what happens to the potato, then you just say what happens. If it says explain, you need to say why that happens. Label with x-axis. Let's label it. Yes, you get a mark for that. It is the mean percentage change and concentration of salt solution. Concentration of salt solution in mole per dm cubed. All right, let's see these answers. Solution was more concentrated outside the potato, so water moves out. One key word you forgot to mention there, osmosis, guys. That's so important. You get a mark for saying osmosis. Someone didn't say that. Got loads of answers to read here. So 3 marks here. If the mass is decreasing, it's losing water, right? So you have to say water moves out of the cells. 1 mark. By osmosis. Why is that happening is the question. Why is that happening? So remember, you got your beaker, you've got your potato inside, you've got your solution. If it's losing mass, what's happening is you have a high water concentration here. You have a low water concentration here. The water is leaving it. So you say that the water concentration in the potato is higher than the water concentration in the solution. Water concentration in the potato is higher than the water concentration in the solution. Are we going through other topics? Yes, we are. So this, for topic 1, these are the main topics that we're going through. So we're doing questions on it. Next, we're going to do topic 2 questions, the key topics on that. If there are any topics that you want to do, like I said, we've got the recording for our boot camp. So like things like stem cells, for example, you can go to that recording, and we've got every topic in there. So if there's like, because we want you guys to practice exam questions with us, if there's any extra that you guys want to go through, then you can watch that recording as well. All right, question on microscope. Name 2 pieces of lab equipment a student could have used to prepare the cells to view using a microscope. What do you guys think there? Yes, it would work for OCR combined higher. We have monoclonal antibodies right at the end, guys, triple guys. Not many triple topics in this paper. It's culturing microorganisms and monoclonal antibodies. Again, it's on the bootcamp recording as well. But at the end, I'll answer a few questions at the end, guys, as well, if you guys have some last minute questions. Yes, so we need a cover slip. Someone said cover slip. There's quite a few things you can say here. Cover slip. What else are we seeing? Someone said slide. Yes. Other things you can mention: slide, cover slip, a stain. Remember that iodine stain? You can say a pipette. That's how you get the water droplet onto the slide as well. Or tweezers if you need to take that layer off the onion cell as well. Yes, someone said tweezers or forceps. Yep, all good, all good. You'll get marks for that. Can you say iodine? You can say iodine. That's all good as well. Part A. Guys, who remembers the name of part A? What's the name? Do not confuse it with this. There are 2 different types of lenses. Yes, eyepiece lens at the top. This one here is the objective lens, okay? Part A is the eyepiece lens. Well done, guys. What is the function of part B? What does part B do? It's not saying name it. What does it do? What do we use it for? So yes, it's the focusing knob. It's used to focus. Yes, to focus. It does technically move the stage up and down as well, but we use it to focus as well. You can also say to improve the resolution of the image as well. Yep. Forceps. Yeah, forceps you can say for that previous question as well. Student tried looking at the cell using a microscope. Suggest one reason why the student could not see any cells when looking through part A. So this student looked through the eyepiece. They couldn't see any cells. Why did they not see a cell? There's like a lot of things you can say here. What do you guys think? Will we do the heart? 100%. We're going to do the heart. It's such a common topic. We're going to do that in the next session. Air bubbles. I like that. You could say there's air bubbles. You could say the light is not switched on. Yes, light's not switched on. You could say the mirror's not in the correct position. You could say the lens was dirty. Microscope wasn't focused. Loads of things we can say there. I'm going to say air bubble because that's the first thing that I saw from you guys as well. Air bubbles. Red blood cells are specialized animal cells. Okay, 6 marker. Let's try the 6 marker, and then I think we can give you guys a break before we go on to topic 2. We've got loads of questions you guys can try after that. 6 marker. Compare the structure of a red blood cell and a plant cell. Red blood cell is unique. We're going to go through this in the next session, What's different about a red blood cell? So we're going to divide it into two things. First, let's talk about the similarities. Because it's a compare, the command word here, compare, we need to say a few advantages, not advantages, a few similarities, a few differences. What are similarities, guys? So everyone's saying no nucleus. That's good. That's the difference. What are the similarities? That's harder to say. I like this, guys. Mitochondria, no. No mitochondria. Cell membrane. Frozen, I see your answer there. It's going. The chat's going. I'm losing track of people. Similarities is cell membranes. Remember I said that every cell has a cell membrane, right? That's the outside part of it, right? Similarities, we can say both have a cell membrane. Is there anything else that both of them have? And yes, you can write in bullet points in your exams. I encourage that. Both have cell membranes. Cytoplasm, both have cytoplasm. Yes, indeed. That's right as well. Both have cytoplasm. There's so many more differences between them as well. Let's talk about the differences. So someone said red blood cell. I'm going to say RBC. You guys don't need to say RBC. You use the full word in your exams, yeah? The RBCs have no nucleus. But plant cells do. Hemoglobin, yes. Red blood cells have hemoglobin. Remember all of this stuff, guys. It's actually the first thing we're going to do in the second session. Our red blood cells have hemoglobin. Plant cells have a vacuole. Yes, they have a permanent vacuole. Plant cells have a permanent vacuole. A permanent vacuole. So guys, you're going to see 6 markers. They have to give you a 6 marker. If your foundation, I think, is 1 6 marker. If you're triple higher, it's 3 6 markers. So don't miss them out. Please don't skip questions. You're guaranteeing 0 marks. Try and just say anything about that topic whatsoever. Write something. You might get a single mark. Okay. I ask this to my students all the time. Do you guys know what an average mark for a 6 marker is in the country? What is the average that you think most of the country get in a 6 marker? Chloroplast, yes. What's the average mark that the country gets in a 6 marker? It's 2. 2 out of 6. It is 2, guys. Think about how low that is, all right? So for you to be above average in the country, you just need to get 3, 1 extra mark, okay? So don't skip these out. Most of the country, most of the people doing GCSEs hate 6 markers. All my students, every time we go through exam papers, they see 6 markers and they want to skip it out, yeah? It's not just you. Everyone wants to do that. If you can just write something, if you can just write a few points and try and get at least 3, you're doing better than most of the country, okay? So just try and write whatever you can on that topic. What else can we say? Hemoglobin, we talk about chlorophyll, chloroplast. Red blood cells are smaller. I don't think you guys really know that. Red blood cells have no cell walls. Yes, plant cells have cell walls. And I think that's enough for 6 marks. And biconcave shape, yes, you can talk about the biconcave shape as well. Right, guys, I think it's time for a break. What do you guys think? We're going to move on to topic 2 next. I'm going to give you guys a 10-minute break. I'm going to move on to topic 2. So in topic 2, we might come back to these questions if we have time in the end. I know topic 3 is very short, so we might come back to these questions if you guys want. We're going to go through blood, heart, human digestive system, plant organ system, food test required practical as well, guys. Can you do 6 markers and bullet points? Yes, you can. Yes, break, guys. We'll be back in 10 minutes. So 6:40, back at 6:40. And we're going to go through topic 2. All right. So go and stretch your legs, or do some more revision if you want. And I will see you guys in about 10 minutes. If you got any questions, yeah, drop it in an email. See you guys in a bit. All right, guys, welcome back. How is everyone finding the session? Are we finding it useful? Let me know in the chat. We're going to go through topic 2. This is by far the biggest topic. This is the one with the most content in it. Yeah? Good, good, good. Minto play, yes. Toby, right? I remember you. Guys, good. It's good to hear you guys are finding it useful. That's the aim here. Remember, I'm not going through every single topic. If you do want to go through all the other topics, we've got the recording for the boot camps. My aim is to get you guys help with the exam session as well. So, yeah, if you want just content, then yeah, there's the boot camp link as well. So, what we have, key topics in Biopaper 1 for topic 2. First thing is blood. Composition of blood. What is blood made out of? We have, let's go to the cheat sheets. 4 components. Here we go. On the far left, we've got the 4 components. Red blood cells, white blood cells, plasma. What do each of these do? And how can you memorize that? Let's go. Red blood cells. So you can see here, they've got 2 O's in it. Yeah, or in other words, you can think of as O2, because what they do is they transport O2. They transport oxygen. Okay, as well as that, they've got a biconcave shape, which increases their surface area. They also have no nucleus, as we mentioned in that question, no nucleus, so that there's more room for oxygen to be carried. Okay, white blood cells also does have an O2 in it, okay? It does have an O2, but this T, if you rotate that, it's like a cross, right? It's got a T in it, so it doesn't carry O2, doesn't. Transport O2. Okay, what does it do? It has an I in it. It fights infection. The I is for infection. It fights infection. And it does that using 3 different methods. We've got phagocytosis, produces antibodies and produces antitoxins. Those we're going to talk about in the topic 3 as well. Then we've got platelets. So for platelets, what do they do? If you want to remember that, there's an S, there's an E, there's an A, there's an L. S-E-A-L. Seal. It seals wounds. We use platelets to seal our wounds. If you get a cut and you want to seal that cut, the platelets come and they cause the blood to clot. They cause blood to clot. Yes, exactly. It is better to say clots blood. Seals is not the best word to use. It's just the word that I could get inside platelets to help you remember that. Yeah, clotting is the better word to use. Plasma, we've got the M, it's for movement, or it moves everything in the blood. It's like the liquid, and it carries everything in the blood. It carries things like, give me examples, guys. What do you think plasma moves around in the blood? What kind of things does it carry? Urea, yes. Ions, red blood cells, yes. Hormones, CO2. One thing you can't say is oxygen, because remember, red blood cells carry oxygen. You can't say oxygen. Hormones, all of those things. Yes. Yes, 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 yes. Okay, cool. Is the 4 different compositions of blood. Okay. Next, we have the heart and the blood vessel. So this is quite a big topic here. You need the structure of the heart. So let's go back to our cheat sheets. Structure of the heart. You need to know all of these labels. Guys, how do you memorize? the order of the circulatory system? I made a video on this like 2 days ago. How do you memorize the order of the... So, yes. Vava, vava, R, vava, vava, L. Yes. So R stands for the right-hand side of the heart. L is for the left side of the heart. There we go. So we have the first V. The first V is for the vena cava. The blood goes in through the vena cava. Then it goes through the atrium. And because we're on the right-hand side, it's the right atrium. Then it goes through the ventricle. And again, it's the right ventricle. Then we got the A. A is for artery because it's going out of something called the pulmonary artery. Pulmonary, the word pulmonary means it's connected to the lungs. So after it goes to the pulmonary artery, it goes to the lungs where it can get oxygenated. So the lungs, you breathe in and all the oxygen from the air goes into the blood, and then it goes to the V. It goes to the left-hand side after that. That first V is for veins. Let me get a different color for the left-hand side. Veins. It is the pulmonary vein because, remember, it's connected to the lungs. Then it goes through the atrium. That's the left atrium this time. Then we've got the ventricle. Left ventricle. And then finally, we have the aorta. Aorta. And then from the aorta, it goes to the rest of the body where the oxygen from the oxygenated blood gets given to your cells so that they can respire. And then they return back to the other side as deoxygenated blood back through the vena cava. Okay? Haven't done the digestive system yet. No, we're going to do that next, actually. So that's the order. So you need to be able to label them everywhere. Okay? So if you've got given a diagram of a heart, then you can quickly write R, Va. No, let me do that again. So this is the right-hand side, R, Vava, and then V, A, V, A, left. R, Vava, Vava, L. Okay? If you quickly just want to remember all the positions of that. Another thing you need to know is where the pacemaker cells are located. So pacemaker cells are cells which control your heartbeat. They're found in the right atrium. Pacemaker cells. Does this work? I don't know. R, A, right atrium. Does that work? It can't be the left. There's no L in it. It can't be ventricle. There's no V in it. Yes. It's found in the right atrium. There's an R and an A in pacemaker. So the pacemaker cells are found in the right atrium. I think only AQA people need to know about the pacemaker cells. Okay? That's a way to remember that. What else do you need to know? You need to know, so we're going to do that. Oh, you need to know about the 3 different blood vessels as well. So we've got 3 different blood vessels: veins, arteries, and capillaries. Here we go. What's the difference between them? Veins have an IN in it. They carry blood into the heart. V, they have valves. What do valves do? They prevent backflow of blood. Why do they have those? Because veins have blood flowing through them at a very, very low pressure. So it's moving very slowly. And if the blood is moving very slowly, there's a chance that it could go in the opposite direction. You don't want that. you want blood to keep flowing in the correct direction. So valves are there. They're like these gates that close if blood tries to flow in the wrong direction. So it prevents backflow. And like I said, the reason for that is because it travels at low pressure. Also, we've got the IN. From the IN, you can also write the word thin, because thin has the IN in it. It has thin walls, muscular walls. Again, that's because blood flows through it at a very low pressure. You don't need thick walls to withstand that pressure. And that means the hole in the middle of it, which we call the lumen. So this is the lumen, the hole is the lumen. This is the muscular wall surrounding it, and these are the valves. Okay, so the hole in the middle, the lumen, is very large in comparison to the wall. So it has a large lumen. So that is the veins. Artery has an A in it. It carries blood away. A for away from the heart. This T, again, we're going to rotate that T to give a cross. It does the opposite. It doesn't have valves. It doesn't travel at low pressure. It travels at high pressure. It doesn't have thin muscular walls. It has thick muscular walls, and it has a small lumen. The cross is to remind you it's the opposite. So we're going to write the opposite of everything we've written. No valves. High pressure. Blood travels at high pressure. Thick muscular walls. And small lumen. Yeah, so because blood is traveling at such a high speed, you don't want the walls to break. So it needs to withstand that pressure. You have this elastic muscular tissue on the walls that prevent it from breaking. They like spring back, basically. Okay. Then we've got the capillaries. Capillaries, the C is to remind you that they connect arteries to veins. C is for connect. So basically, blood leaves the heart through arteries, then it goes through capillaries, where it goes to every single cell in the body, and it transports the oxygen to it. Then when it's done, all the capillaries come together to form a vein, and then the veins take it back to the heart. Okay, so they're basically connecting the arteries to the veins. The P there is for permeable. Permeable, what does that mean? Permeable means it lets substance through it. The walls are permeable because you need to transport substances. So you need to diffuse substances into and out of the capillaries. When blood goes up to like a cell, for example, it needs to give the cell oxygen, and then it needs to take away the waste products like carbon dioxide. So the walls have to be permeable. So permeable walls to allow substances to diffuse in and out. The C as well, you can also use the C to get the word cell. That reminds you that they are one cell thick. Capillaries are, the walls are one cell thick. And again, that's to make it easier for the substances to diffuse in and out of them. Yes. Well done, guys. Well done. So that covers blood and the heart and blood vessels. So 3 key topics there. Comes up loads and loads and loads. Very common topics. Next, we've got the human digestive system. So the order of the human digestive system. let's look at a diagram for this. Oh, by the way, you do get asked about the lungs sometimes as well, not that much. So you do need to know about the lungs. You need to know about the trachea separating into the bronchi, and then separating into bronchioles before going to the alveoli. This is how we get oxygen from the air into our blood. We use the lungs, and we've talked about alveoli already. Cardiovascular disease will come. I just wanted to go through the digestive system first. Okay, so digestive system, we're going to talk about the 3 types of enzymes you can have here. Okay, so enzymes are used in the digestive system to break down certain types of food. So enzymes are biological catalysts. And there's 3 types of digestive enzymes we have. And you can easily memorize what each one breaks down because they're similar to the names. So carbohydrates, protease, and lipase. Those are the 3 types of enzymes you can have. Carbohydrates breaks down carbohydrates. Protease breaks down proteins. Lipase breaks down lipids. In other words, lipids are fats as well. What do they break them down into? Carbohydrates get broken down into simple sugars. Proteins get broken down into amino acids, and fats get broken down into 2 things, glycerol and fatty acids. Some of the examples you might have heard of, I think the one that you hear the most is amylase. Amylase is an example of a carbohydrate. It breaks down carbohydrates. It breaks down starch in particular. You can find it in your saliva in the mouth as well. So where do you find all digestive enzymes? This is the order of the digestive system. We use muscle pair to remember. Muscle pair. So mouth, esophagus, then it goes to the stomach, then it goes past the liver, then it goes past the pancreas, then it goes through the intestine, the small intestine, then the large intestine, and then finally out of the rectum. Okay, so let's just write that down on our notes as well. Down here, muscle pair, just to remember the order. So we've got mouth. So food goes into the mouth. You chew it. In the mouth, you also have amylase, which breaks down starch in the saliva. Chewing the food is mechanical digestion. Then you swallow your food and it goes down the esophagus. Not much to know about the esophagus. The esophagus is just this long tube that connects the mouth to the stomach. Stomach, you need to know quite a lot about. Stomach, it produces hydrochloric acid, HCl. That is there to kill bacteria. And also, it's there to provide the correct pH for enzymes. Loads of people think it's there to break down food. The hydrochloric acid is not there to break down food. You don't say that in your exams. You say one of 2 things. It's there to kill the bacteria, or is to provide the correct pH for enzymes to work. Because in the stomach, you have protease enzymes. Remember, the protease enzymes are the ones that break down proteins, and they work the best in acidic conditions. So they work the best when the pH is like around 2 or 3. So the stomach has to be acidic so that those enzymes work the best. Yeah. Also, the stomach has walls which churn the food. I think someone said that. Yes, that's correct. EdXL guys don't need to know. I'm pretty sure you don't need to know the digestive system. You need to know the digestive enzymes, but you don't need to know the system itself. Then we've got the liver. The liver produces something. Can anyone tell me what the liver produces? What does the liver produce? And loads of people think it's an enzyme. This thing is not an enzyme. It does work like an enzyme a bit. There we go. Everyone's saying it. Bile. Yes. Bile is not an enzyme. Okay. Let me get a different color for this. Bile is not an enzyme. It's just a substance which breaks down fats into tiny droplets. Remember, enzymes break them down into other chemicals. So like if it was a lipase enzyme, it would break it down. It break the fats down into glycerol fatty acids. The bile only breaks the fats down into smaller fat droplets. So it's not an enzyme. We say it emulsifies fats into smaller droplets. You basically want to do that so that it increases the surface area of the fats, so that then lipase enzyme can break it down even further after that as well. Not only does it do that, bile also, bile is alkali. So remember, in the stomach, you have acid, right? So all your food has just gone into your stomach, it's turned acidic, and it's come out of your stomach, and now you've got acid in your body. You don't want acid to go into the rest of your organs. You don't want it to go into your intestine. So the bile itself is alkali. It neutralizes the acid. So it neutralizes stomach acid. Then we've got the P. The P is for pancreas. Pancreas. What does the pancreas do? The pancreas produces all three types of enzymes. So carbohydrates, protease, and lipase. Very simple. Then we've got the intestine, the small intestine first. The small intestine is where food is absorbed from your body into the blood. And also, you've got all three enzymes as well. Let me put an arrow here. It produces all three enzymes: carbohydrates, lipase, and protease. Then we've got the large intestine. The large intestine is there to absorb excess water. So you have some water left over, and you need to absorb that as well. The large intestine does that. And finally, you've got the rectum, which stores all the waste as feces. There we go. Okay, next we've got human digestive system done. Plant organ systems. So let's look at what we need to know for plant organ systems. So digestive system done. We'll come back to the second. Plant organ systems. You get loads of questions asking you about the phloem and the xylem. That comes up in this topic. It also comes up in the specialized cell topic as well. It's quite a common question. What's the difference between the phloem and the xylem? Well, one transports sugars around the plant, and the other transports water around the plant. Okay. This one, the phloem, is made out of living cells. The xylem is made out of dead cells. Because it's dead cells, it means it has to be strengthened, because if the cells are dead, it's not going to be very strong. So it's strengthened by a substance called lignin. Another few differences between them. Phloem cells have like these plates between them. Whereas if you look at a xylem cell, you can see it's completely hollow. It's completely hollow. There's no plates. There's no end wall plates here. And the direction is another difference. Xylem cells, xylem tubes, always take water and minerals up the plant. It never goes in any other direction. It always goes up. Whereas phloem tubes can take sugars up and down the plant. Okay, so the process of sugars moving around the plant is called translocation. And then the route of the xylem is called the transpiration stream. Transpiration stream, right? We've got a memory trick for you guys about this as well. Phloem and xylem. So I tell people to write phloem with a tick as the L, the reverse tick as the L, and a cross for xylem as the X, and we write let. Each letter in let stands for something. Living cells, end walls, and two-way flow. Two-way flow. Okay, so in phloem, all of the statements in let are true. So they are made of living cells. They do have end walls between their cells. Remember I told you they have those end plates, and they do carry sugars two ways, upwards and downwards. Xylem does not. It's not made up of living cells. It's made out of dead cells. It doesn't have end walls, and it doesn't have two-way flow. So it's made out of dead cells. Remember, it's strengthened with lignin. Makes it stronger. No end walls. It's completely hollow. And only one-way flow, so water only moves upwards. It's come up as a 6 marker, guys, before. Differences between the xylem and the phloem. Imagine that comes up tomorrow. You guys will get 6 out of 6 there. Those are the differences between them. Okay. What else do we need to know? So those are the ones that get asked the most. We've talked about root hair cells already, kind of. We talked about the fact that it absorbs water, right? It absorbs water by osmosis. It also takes in mineral ions. What does it use to take in mineral ions, guys? Let me know. Let me know in the chat. So it takes in water by osmosis. It doesn't take in mineral ions by osmosis. And people always forget that. Who knows? There we go. Active transport, yes. So mineral ions, it needs to take from the soil. It does that by active transport. The reason for that, guys, is because in the soil, the concentration of ions outside is much lower than inside the root hair cell. So you only have like a few mineral ions outside, whereas inside you have loads of mineral ions. So you have a high concentration inside the cell, you have a low concentration outside the cell. So you need to go against the concentration gradient to take those mineral ions in. And that process, remember, needs energy. That's active transport. Then another thing about root hair cells is that they've got those long hairs, those long projections, and that increases their surface area. Okay, then we've got stomata. So stomata are these holes that you find in leaves. Which part of the leaf, guys, has the most stomata? Which one has the most holes in it? What do you guys know? Which part of the leaf has the most holes in it? So they have these holes and they're surrounded by cells called guard cells. It's the bottom bit. Is the bottom bit, the lower epidermis. Yes, exactly. The bottom, if you think of a leaf, the top bit is getting the most sunlight, right? So near the top, we have the palisade mesophyll layer. That's where all the most chloroplasts are. At the bottom, you have a lot of stomata, a lot of holes, because that's the shaded side. It doesn't need light. So what happens there is gas exchange. It takes in carbon dioxide for photosynthesis. It gives out oxygen from photosynthesis as well. And what these guard cells do is they open and they close based on whether photosynthesis is occurring or not. So in the daytime, there's a lot of sunlight, a lot of photosynthesis is going to happen. So these guard cells open the stomata because it needs to take in a lot of carbon dioxide to make food for the plant, the glucose. But at nighttime, it doesn't need to do that. There's a problem. At nighttime, because all of these holes are there, all of these stomata are there, the plant can lose water and it doesn't want to lose too much water. The plant needs water. So at nighttime, these stomata close so that water can't leave. And because it doesn't need as much CO2 because it's not photosynthesizing, it might as well just close the guard cells. So that's the information that we have here as well. Okay. So transpiration. So we talked about the transpiration stream. The transpiration itself is in the leaf. I told you it loses water through the stomata. Transpiration is caused by evaporation and diffusion of water from the plant's surface. Normally, it mostly happens in the leaves because it has all of these stomata. So this is the full transpiration stream, guys. Water goes into the leaves, or into the roots, sorry, by osmosis. Remember, the root hair cells takes it in by osmosis. Then it goes up the plant through the xylem. Then it goes to the leaves. And through the leaves in the stomata, it goes out by evaporation. Okay, so I've seen like a 4 marker on this. And yeah, I tell people to use Rooksets to remember that. So R is for root hair cells. So the plant takes in water through the root hair cells. It takes it in by osmosis. It takes it up the plant through the xylem, and then evaporation occurs. Or in other words, transpiration occurs through the stomata, those holes that we were talking about earlier. All right. Now, transpiration can be increased. The rate of transpiration, so the rate at which these plants lose water, can be increased by 4 different factors. 4 factors that affect transpiration, guys. What do you think they are? 4 factors. Let me know in the chat. How can you make plants lose water faster? Halt. Yassine said halt. Yes, you know the memory trick. Why is people saying halt? Not licut. That is for photosynthesis. I'll talk about that later. Halt. What does that stand for? Those are the 4 factors. Humidity. I'm seeing all the correct answers, guys. Well done. Airflow rate. Light intensity. And temperature. Okay. 3 of these. So temperature, if you increase temperature, transpiration is faster. You're going to evaporate more water, right? That makes sense. If you heat something up, more water is going to evaporate. Light intensity. If you increase light intensity, that's going to cause transpiration to increase as well. Because remember what happens when sunlight is bright? The stomata open up. The guard cells open up because they basically become full of water. They become turgid. So that means it's going to increase the amount of water being lost. Airflow rate, wind speed, that's basically wind speed. The faster wind is flowing, the faster water is lost. That's just because of the concentration gradient. If it's blowing wind away from the surface of the plant, it's keeping the concentration of water very low outside the plant, so it leaves faster. Humidity, if you increase humidity, that's the one factor that causes transpiration to decrease. Okay, so if you increase any of these, the last 3 will cause transpiration to increase, but humidity causes transpiration to go down if you make it more humid. Humid is, think of it as how much water concentration there is outside the plant. If you have a lot of water outside the plant, then diffusion of water is going to be slower because the concentration gradient is going to be lower. So that's the only one that causes it to decrease. So remember halt for all of those factors. Okay. Right, let's go back to our list, see what we've got left. Was that it? So we've gone through human digestive system, plant organ system. How are we doing for time? Okay, we're doing good. We're doing good. One more required practical food test, guys. 4 food tests. Let me know. What do you know? We've got. Let me know the food tests that you guys know about. What's Rooksets for? Rooksets is for the direction of water flow in a plant, in the transpiration stream. Benedict's, okay. What do we need to know about Benedict's? So for Benedict's, is a test for sugar. Yes, test for sugar. This is the one you need to heat up, guys. Heat to around 65, or let's say 70 degrees C in a water bath. Color change. Color change. What's the color change for the Benedict's solution? Or here. Okay, so I always say brick red, but you can say yellow, you can say orange, you can say green, you can say loads of different colors for this one. Okay, so it goes blue to default. I always say brick red. Yeah, but if it changes any other color from blue, it means sugars are present. Memory trick. Ben likes bread. How does that work? Green, yellow, and brick red. Yes. Then we've got Biuret. People get these two mixed up. Biuret, this tests for proteins. Proteins. Color change, blue to purple. The two that begin with B start off as blue. So Benedict's and Biuret starts off with B. They start off as blue for B. For this one, you usually need to grind up the food. If it's a 1 marker, you don't need to mention that. But if they ask you like a 3 marker, mention that you need to grind up the food. Yeah. And same with Benedict's. Benedict's, if it's a 1 marker, you don't need to say heat it up. But if it's more marks, then say heat it up as well. Then we've got iodine. Iodine tests for starch. Iodine test for starch. Color change for iodine, guys. What is the color change for iodine? It goes to blue-black. Yes. What does it start off as? Orange. Yes. Orange or brown. You can say orange or brown. You get the mark for either one. So I'm going to put orange/brown to blue-black. And the one that I have surprisingly, correct me if I'm wrong, in AQA, I have never seen this come up. I've never seen the lipids one come up, the emulsion test, which is basically ethanol. I've never seen it in an actual past paper come up. It's the test for fats. What do we see here? It goes cloudy. Yes. You shake it. You can use Sudan 3 as well. Yes. So it goes from colorless, we'll say clear colorless, to cloudy. If you do talk about Sudan 3, yeah, it forms a bright red layer in the test tube. So you can mention that as well. Okay, so those are the 4 food tests. Sudan 3, again, the test for lipids, I haven't seen come up. It's always the top 3 that comes up. But anyway, it's just one extra one to learn. Okay, I think we're ready to do some questions. Let's try some questions now. I think we're doing good for time. Okay, I'll give you guys a break soon. Straight in 4 marker on food tests. Guys, let's see. 4 marks for this. Describe the chemical tests that students could show if a bread contains starch or iodine. Sorry, starch or sugar. I've given away the answer there. So for starch, so like I say, guys, use bullet points for 4 markers. You can say iodine. What was 35 degrees? I'm not sure. 35 degrees. What do we say 35 degrees for? Benedict's and iodine. Yes. For starch, add iodine. Iodine solution. Yep. And then say the color change. Say what it starts off as and what it ends up as well. Okay. So it goes from orange to colorless. Look at the question carefully as well. Sorry, not colorless. Sorry, orange to blue-black. If starch present. Yeah. It says in the question, you should include the results of a positive test and a negative test. So we do need to also say what would happen if there was no starch. What would you say for that? What would you say if there was no starch? What would you say? If not, no color change. Yes, exactly. Yeah. So you can say remains orange if no starch present. Okay, let's do the same now for the sugar. What are we going to say for sugars, guys? For sugars, we're going to use, yes, Benedict's. Remember, what else do we need to say for Benedict's? Because it's a 4 marker, we need to give a bit more information. 75 degrees C. I'm looking at the mark scheme right now. It says at least 60 degrees. So you have to say a temperature higher than 60 degrees. If you say a temperature lower, you're not going to get the mark. So add Benedict solution. Heat to, what do we say? 75 degrees C. Yes. Color change. Color change. So if there is sugars present, you'd say it goes from blue to, like I said, default, I say brick red, but you guys can say anything. If sugar's present, and then it remains blue if no sugar is present. Don't say brick red precipitate. No, just say color change. Yeah, you don't need to say precipitate. Precipitate, you say more in chemistry. In chemistry. Nice. Well done, guys. Straight in with the fucker there. A student investigated 3 types of bread. For each type of bread, the student put a square piece of bread into their mouth. Okay. They did not chew the bread, and they recorded the time for the bread to taste sweet. So people get confused here and wonder what the hell's going on here. Think about it. In your mouth, remember what you have in your mouth. You have amylase, right? Amylase breaks down starch. Breaks down starch. It's a carbohydrate enzyme. It breaks it down into sugars. As soon as you get sugars, it's going to start tasting sweet. So initially, bread doesn't taste sweet, right? But if you keep it in your mouth, it eventually breaks down into sugar and it starts to taste sweet. So what they're doing is they're putting different types of bread into their mouth and timing how long it takes for it to turn sweet, because you're basically assuming the moment it turns sweet is the moment sugar is produced. And this is what this question is all about. It says the starch in the bread was broken down by the enzyme, what we're going to say here, amylase. Yes, someone said it. Amylase. The enzyme broke down the starch into, what did it break it down into? Sugar. Yes, sugars, simple sugars, sugar. Nice. What was the independent variable in this investigation? Independent variable. Remember, independent is for what you are adjusting, changing. The type of bread or the size of the bread, I would say this is the control variable. Remember, they're putting a square piece of bread into the mouth. You want to keep that the same. You don't want different you don't want different amounts of bread. Otherwise, they're going to take a different amount of time to break down. Temperature of the mouth, you want to make that a control variable. Type of bread is what they're changing. They're testing different types of bread. That's the independent variable, right? What's the dependent variable, guys? Tell me that. What's the dependent? So this is independent variable. What's the dependent variable? Yeah, this is for everyone, guys. Everyone needs to know this. The time. Yeah, we're measuring time. The time for it to turn sweet. So time to turn sweet is the dependent variable. What are 2 conclusions that we can make from the results? So look at this table, guys. 2 conclusions that we can make here. Let me know. 2 marks, 2 points, 2 conclusions we can make. White bread turns sweeter the fastest. Yes, we can say that. I think that's a good thing to say. And wholemeal takes the longest time. Yep, exactly. So white bread takes the shortest time to turn sweet. Wholemeal bread takes the longest to turn sweet. Foundation, yes, this can come up in foundation. This can come up in higher as well. It's relevant for everyone. So the table is repeated. So this is the same table we saw earlier. The student improved the investigation. Okay, so look here, we can see a slightly different table, a bit more information in it. What have they done to improve the investigation? Looking at this new table, what would you guys say? And this is something that comes up a lot. When they ask you, how can you improve an investigation? You usually say these two things. There's two things. Sometimes it's 1 mark, sometimes it's 2 marks. So saying created a mean or added a mean is not enough. You have to say repeat and calculate a mean. If you get a question asking how to improve something, you can't just say calculate a mean. You won't get all the marks. Every time you look at a mark scheme, it will say repeat and calculate a mean. The and will be in bold letters, saying meaning you have to say that to get both marks. So what did they do? They repeated investigation and calculated a mean. Calculate a value for x in table 3. We just need to calculate a mean, guys. Very common maths calculation. Yep. Add them all up, divided by 3. Has anyone got an answer? 58. Yes. Well done, guys. So it was 58 plus 55 plus 61 divided by 3 gives you 58 seconds. Why should the student investigate with more people? Which of these would be the answers here? Why should the student investigate with more people? Everyone point A. Each person's sense of taste is different. Yes, I would say that's right. They have to decide when they taste something sweet, right? Some people will taste it sweeter earlier. Some people would sense it later. More people will make it safer. Not really. You're doing the same thing every time. There are many types of bread. That's irrelevant. That doesn't mean you need to test more people, because one person is testing. The whole point is one person is testing loads of different types of bread. So having more people is not going to change that fact. Ooh, heart question. Heart question. Very common question here. If you see a diagram of the heart, remember R, VAR, VAR, L. So we've got right hand side on the left hand side on the right. Remember to switch them around. So R, VAR, L. Okay. Quick way, if you guys just want to quickly remember. Which part of the heart receives oxygenated blood from the lungs? Guys, which one is it? Let's remember the order. So R, VAR, then it goes to the lungs. Then it goes L to the left-hand side, VAR, and then out to the rest of the body. So which one will it be? We've got some people saying A, we've got some people saying B, we've got some people saying D. No one said C. It's definitely not C. Is it A? I think it is. Because remember what's happening? It goes R, VAR, then it goes to the lungs. What does the lungs do? It oxygenates the blood. You take a breath in, oxygen goes into your blood goes in this way into A, the left atrium. So that receives the oxygenated blood first. Which part of the heart pumps deoxygenated blood to the lungs? A, B, C, or D? What would you guys say it is? A, B, C, or D? Pumps means which one is the one that's pushing the blood out? It says to the lungs. So just like before, remember I said the lungs are here. Then it goes back into the heart this way. We've got most people saying C. C is correct. C is the right ventricle, V for ventricle. Ventricle pushes the blood out of the pulmonary artery to the lungs in order to be oxygenated. The answer there is C. Well done, guys. You're smashing it. Spin the wheel. No, no, we're not doing spin the wheel. This is pure revision today. A group of cells called the pacemaker controls the resting heart rate. Where's the pacemaker found? Remember, guys, if you want to remember it, pacemaker has an R and an A for right atrium. Right atrium, right atrium. There's no L. There's no V for ventricle. So it's not ventricle. There's no L. It's the right atrium. Pacemaker has an R and an E. R and an A in it. Next one. Figure 2 shows a cross-section of an artery and a vein. Describe 2 ways the structure of an artery is different from the structure of a vein. Guys, what can we say here? They've given us a diagram, but remember, we've got the V, we've got the IN at the end for one, and then we've got the artery, which is A. We've got the cross in the middle. Artery. Thick wall, narrow lumen. Yes, yes, yes. Okay. This is very important, guys. Let me see what you guys are saying. So some people are saying thick walls, thick elastic tissue. You don't get a mark for that. You have to say thicker. You're comparing. It says describe. You're comparing 2 different things. You have to say one has a thicker muscular wall. Okay. So we can say arteries have thicker muscular walls. And then we can say narrower lumen. One more thing we can say. Let's talk about arteries. Arteries have narrower lumen. Valves, yes. Arteries do not have valves, so you can say that as well for the marks. Amazing. Coronary heart disease. We're going to talk about coronary heart disease later. Let's see what else we've got here. Okay, let's do this plant question. We might come back to the one before because I think in the next session we're doing coronary heart disease. It's technically a topic 2 topic, but next session doesn't have that many topics. It's the shortest one. It's on communicable diseases. So maybe we can come back to that question. So let's go to question 4. What is the process by which water is lost from the leaves in a plant? No, not osmosis. Not osmosis. It's lost from the leaves in the plant, guys. It's transpiration. Osmosis is how it's taken in through the roots, root hair cells, remember? Photosynthesis, no, that's the process by which plants make their own food. That's not it either. The answer here is transpiration. Here, look, that's the diagram I showed you earlier. We've got the stomata. What are the 2 cells that surround the stomata core? Cell X, is that the next question? Yeah, what's the name of cell X, guys? Guard cells. It's a mix of all types of papers, guys. If something's higher only or triple only, I'll tell you if it's triple only. So far, this is all relevant to everyone. Next, we have a calculation question here, a magnification question. They're being nice. They've given us an equation here. But if you weren't given the equation, remember, I am to remember the equation. So magnification is equal to image size over actual size. The question is asking, calculate the real length. So they have given us the length of the image in the microscope, and we've got a magnification here as well. We want the actual size. So we want the A. A is equal to I over M. So what do we need to do here, guys? Actual size is equal to image size over magnification. That's what they've given us here, but I'm assuming we didn't get that equation, just in case tomorrow you guys don't get given the equation. We want to do the 25, yes, over, so 25 over the 800, the magnification. Is that the final thing to do, guys? Look carefully. Look at the question carefully. So loads of people have said yes. Okay, people have done it. The thing you need to do at the end. So doing 25 over 800, that gives you 0.03125. Okay, but that's millimeters, because you got to remember in the question at the top here is 25 millimeters. The question clearly says give your answer in micrometers. We need to convert from millimeters to micrometers. How do we do that? If you guys remember from the first session, you need to times by 1,000. If you want to go the other way, you divide by 1,000. So let's get that number and times it by 1,000. Let's move the decimal point 3 places. 1, 2, 3. 31.25. I think that's good enough. 31.25 micrometers. Okay. Cool. Next, the student measured the mass of water lost. Okay, so this transpiration question, water loss is transpiration. From some similar plants, the plants were at different temperatures. This is the apparatus. So they see the plastic bag around the pot of a potted plant. So they've got a plastic bag here. I think there's a question asking why. We'll answer that in a second. Place the potted plant with the bag on a balance in a room at 20 degrees C. Okay. So they're noting down the mass here. Record the mass at the start. Then wait 3 hours and record the mass again. So what's probably going to happen is the mass is going to go down because the plant is losing water. And repeat it at different temperatures. So this is an experiment that's investigating transpiration at different temperatures. So remember the factors affecting transpiration? There's 4. HALT, H-A-L-T. The T is for temperature. What was the independent variable here? What's the independent variable? What are we changing? E, not E. More 6 markers. I think we've got a few 6 markers coming up. Don't worry. Temperature of the room. Yes, that is what we're switching. That's what we're changing, right? So temperature would be the answer there. Initial mass of the plant. No. Length of time. That's probably the control variable. Mass of water lost. That's what we're measuring. So that's dependent. That's independent. Suggest why the students sealed the plastic bag around the pot. Okay, here we go. Why do you think this plastic bag was here, guys? Why have we done that? Investigating transpiration. Why do you want to wrap the bottom of the plant with a plastic bag? When is break? I'll give you a break in 5 minutes. Because it's the biggest topic, I want to spend a bit longer on this topic. Traps heat? No. Reduces humidity? No. To prevent water loss. We're trying to measure water loss. So saying that is not enough, but very close. You can't just say prevent water loss. To prevent water loss from the rest of the plant. You see here, there's soil, right? There's water in the soil. Water could evaporate from here as well. You don't want to measure that. You're trying to measure water loss from the plant only water from the leaves. Yeah. So you don't want water to be lost from the soil. Otherwise, that's going to make your investigation unfair. So to prevent water lost from the soil. Yes. Not trap oxygen. No, that won't get you a mark. Okay, next one. We've got the results here. What's the resolution? Ooh, okay. What's the resolution here, guys, of the balance? Resolution is the smallest value that the balance can read. And you can tell that by how many decimal points there are here. 0.1 is correct because it's all 1 dp, right? So think about if you had it at the very smallest, if you had no mass at all, would the balance read? It would read 0.0, right? The smallest reading it can read is 0.000.1. That's the smallest value that the balance can read. So the answer is 0.1 grams. That's the resolution. Okay. Barely gets asked, by the way. But if it does, you'll get that one, you know. Calculate the value of X. How do we calculate the value of X here, guys? How do we calculate the value of X here? Look at this table carefully. We've got the mass at the start. We've got the mass at the end after the 3 hours. And then we've got the mass of water lost. People are telling me the answer already. Yes. Basically, if you take these two values away, you get the final answer, right? Minus that gets you that. So something minus 506.3 gives you 4.9. So we just need to do 506.3 plus 4.9. 506.3 plus 4.9. And that will give you, like you guys are telling me, 511.2 grams. Yes, we're going through diseases straight after the break. Don't worry. Don't worry. One, what can we say here, guys? We've got temperature 30 degrees. What can we say here? Where can I get the paper? Guys, remember, we can send this to you. If you email us, we'll send you the sheet. You should have got it sent if you did sign up. Okay. Conclusion is a higher the temperature, the more water loss. If there's lag, guys, sorry. It does stabilize after a bit as well. Can you guys hear me okay, though? Is the sound okay, or is that messing up as well? Sounds okay. Okay, so we're going to go through, we're going to have a break now anyway. So hopefully by the end of the break, it'll be fine. Give 2 factors that might affect the rate of water loss from leaves. Guys, remember, what's the memory trick here? What can we say? 2 things we can say. It says, do not refer to temperature. Anything in halt will do. Not stomata closing. I wouldn't say that. It might get you a mark. But yeah, let's say humidity. H for humidity. A for airflow rate. Or light intensity, if you want as well. Any of those would get you. Okay, we're done here. Oh, actually, we skipped one, didn't we? We'll come back to this actually after. I think we got a nice 6 marker there as well. Yeah, we can try the 6 marker. We can have a break, guys. We're going to have a 10-minute break now. So, yeah, go stretch your legs or something. Come back in about 10 minutes. Let's say at 7:55. 7:55. We'll be back. And we'll do topic 3, which is all about diseases. And we're going to talk about communicable and non-communicable diseases and then try some questions on that. We're about halfway here, guys. Okay, you guys are doing really well, smashing it. I'll see you guys in about 10 minutes. See you then. All right, guys, welcome back. Welcome back. Hope you guys are finding this useful. Halfway point past, we're on topic 3 now. So topic 3 is communicable diseases. So it's the shortest one. What I'm going to do is I'm going to try and finish this one early so that in the final session, the main one, anaerobic respiration and photosynthesis, I want to spend the most time on because those are huge topics, very important topics. And remember, those are the ones that can come up in paper 2 as well. Okay, those like get asked so much. All right, so we've got to do communicable diseases, and also we're going to go back to topic 2 and do non-communicable diseases. We're going to look at coronary heart disease as well. 9:30 ending. That's the plan. Okay. Hello, everyone. Hope you're all doing well and you're keeping up. You're keeping up with me. Okay, so communicable diseases. We're going to start with 4 types of pathogens you can have. All right, 4 types of pathogens. Most of the ones they ask you about are bacteria and viruses. So bacteria, they're living cells, very small prokaryotic cells. We've mentioned that already right at the start of the live. So that means they don't have any nucleus. They have all of their DNA floating around in the cytoplasm. And the way that they make us feel ill is they produce poisons called toxins. Okay, so remember, we're going through pathogens right now. So 4 types of pathogens. Bacteria is one of them. If you get asked what is a pathogen, you'd say it's a microorganism that can invade and harm the body. All right, so how does bacteria harm the body? It produces these poisons called toxins. None of the other pathogens cause toxins. Viruses, they harm you a different way. They're technically not cells. So what they do is they go inside and reproduce inside a body cell. Okay, so they like attach to the surface of a body cell, and then they start reproducing inside, and eventually they reproduce so much that the cell bursts. And that cell bursting is what causes the person to feel ill if they've got a viral disease. The cell is what causes that to happen. Then we've really come up when you're talking about the disease malaria. And they're eukaryotes, so they do have a nucleus, usually single-celled, and they live as parasites. So we have parasites because what parasites do is they live inside an organism or on an organism, and then they cause that organism to harm. The way that protists are spread, so these communicable diseases means the pathogens can spread from one person to another. The way that they spread is using a vector. So a vector is just like an organism which transfers the protist, but it doesn't get ill itself. So like a mosquito, for example, transfers the malaria protist, but when it's transferring it, when it's biting one person and then goes to bite another, the mosquito itself doesn't get malaria. So we call the mosquito a vector because it's carrying the disease, but not getting the disease itself. Okay. Fungi is the last one. They get asked the least. There's only one disease that you need to know for fungi, which is rose black spot. It's one based on plants, and they're usually multi-celled, and they reproduce using these structures called hyphae, and they produce spores. Doesn't really get asked, so I'm not going to go through that properly right now. All the diseases that you need to know. So if you're AQA, these are the diseases that you need to know. If you're Edexcel or OCR, it's going to be slightly different. Okay, so the viral ones, the ones caused by viruses, are the top 3: measles, HIV, TMV. You need to know how they're spread, their symptoms, and how to treat them. Okay, so let me quickly run through them. So measles, think of COVID. They're spread by coughing and sneezing. They cause a red skin rash. How to treat them? There's a vaccination for measles. It's called the MMR vaccine. MMR and rubella. I've seen that asked before. They've asked for a 1 marker, what does MMR stand for? So remember that. And that's how you prevent it as well. HIV is a sexually transmitted disease. So it can be spread through sexual contact, but also through blood as well. Symptoms, it starts off as flu-like symptoms and it leads to AIDS. AIDS is when your immune system is basically killed. So you have no white blood cells to fight off diseases. And it leaves you prone to being attacked by other diseases as well. How to treat it? We have these drugs called antiretroviral drugs, which kill them as well. You can prevent it by using like a condom, basically, and no sharing needles. TMV and rose black spot, these are both plant diseases. So TMV is a virus, and that causes discolored leaves and stunted growth. Why does discolored leaf lead to stunted growth? This gets asked quite a lot. Imagine you have a leaf that's not green. What makes a leaf green? It's the fact that it has chlorophyll in it, right? Chlorophyll absorbs sunlight. If it's discolored, it doesn't have any chlorophyll in it. That means it's not going to be able to photosynthesize, which means it's not going to be able to grow. Therefore, that leads to stunted growth. There's not really any cure. You can just prevent it by preventing contact of plants. Then we've got rose black spot, which causes purple/black. And again, it discolors the leaves as well, prevents. You can with fungicide cream, which basically kills the fungus. Malaria is the one that gets asked the most, guys. Malaria, and this is all exam boards ask malaria. Malaria is caused by a protist, and like I said, it's mosquito bites. The mosquitoes are the vectors. They carry the protist from one person to another. If you get malaria, you get fevers, so repeating episodes of fevers. You get chills, and you get very tired as well. You can get drugs to treat it. You can have a vaccination as well, or you can control the mosquitoes. You can use nets, you can use insecticide to prevent them as well. Okay, then we've got two bacterial diseases as well, salmonella and gonorrhea. So salmonella, that's food poisoning. It's caused by contaminated food, specifically from poultry, from like chicken. It can lead to fevers, stomach cramps, vomiting, diarrhea. Because it's bacterial, you can use antibiotics. Okay, so antibiotics can be used against bacterial diseases only. You can't use them against viral diseases. And to prevent them, you need good food hygiene, wash your hands, cook the food thoroughly, the chicken thoroughly. And then gonorrhea is another sexually transmitted disease caused by a bacteria. Painful urination, genital discharge as well. You can treat it with antibiotics as well, and also safe sex, so condoms as well. Okay, defense systems, human defense systems. How do we prevent diseases? How do we prevent things entering our body? You can use SNASI here. SNASI is the memory trick here. SNASI. So we've got the skin. The skin acts as a barrier to prevent microorganisms entering the body, pathogens from entering the body. And also it produces these oils which kill the pathogens. In your nose, you have hairs and mucus. So skin, I'm going to write barrier. In the nose, we got hairs and mucus, which trap pathogens, stop them from going further into your air passages, which is the next one. In your air passages, you have hairs again, but they're not called hairs. So it produces mucus, but instead of hairs, does anyone know what it has? What do the air passages have? Let me know in the chat if you know what they have. So this is the trachea and the bronchi. What do they have? They have cilia. Well done, guys. Cilia cells. Cilia cells are hair-like structures. They're not hairs. They look like hairs, but they can move. So they can move up and down. So if you have mucus, which has the pathogen trapped within it, the cilia can waft the mucus. It can push it back up your trachea to the back of your throat, where you can basically swallow it. And then it'll get destroyed in the stomach, or the pathogens will get destroyed in the stomach. And that's the S here, the stomach. The stomach has hydrochloric acid in it. It's got acid in it. And you remember from the digestive system, hydrochloric acid kills bacteria. Finally, if all of that fails and you get infected, this is where you get the immune system. And the immune system kicks in once you've been infected. How does the immune system help you against an infection? 3 things. APA. Okay. First one is antitoxins. It produces antitoxins. Antitoxins, remember I talked about bacteria like 5 minutes ago? Bacteria produces poisons called toxins. Antitoxins are against toxins, so they neutralize those toxins. So it gets rid of them. So that helps you. It makes you feel better if you've got a bacterial infection. Second one, phagocytosis. So you've got these white blood cells called phagocytes. They engulf and digest a bacteria or any pathogen, meaning they can't harm you. And finally, we got antibodies. So antibodies are also produced by white blood cells. Specifically, they're called lymphocytes. And the antibodies, they attach onto the surface of a pathogen. So on the surface of a pathogen, you have these markers called antigens. And they attach onto the antigens. They make the pathogens clump together. And then the phagocytes come and engulf the pathogens. After you have fought off an infection, right? You've killed off most of the pathogens, or you killed off all the pathogens. So imagine you've just had, like, I don't know, chickenpox or something. Your white blood cell have just killed the whole virus, every single virus in your body, okay? So all of these white blood cells, which produced the antibodies, they're there in your body, but you don't need them anymore. So they basically start dying off. But not all of them die off. There'll still be some left behind. Some of these lymphocytes that have produced the antibodies to kill the chickenpox virus stay behind. And at that point, they stay behind as memory cells. We call them memory cells. So memory cells are basically white blood cells or lymphocytes that have stayed behind after an infection. So that if that same pathogen was to come into your body again, the memory cell will know what antibody to produce to kill that pathogen, because it has already fought off the chickenpox virus. So if that same virus comes in again, those same lymphocytes will just rapidly produce the antibodies to instantly kill it before it gets a chance to kill you, not kill you, to harm you. So at that point, you say that the person is immune to that disease. Okay. Okay, so that is a memory cell. Next up, let's see what else there is on the list. So like I said, this one's got a shorter one. We're going to go through coronary heart disease. I'm not frozen, right? No, okay, fine. Am I frozen, guys? Let me know. I need to do something if it is. I'm not, okay. Okay, so cardiovascular disease. So we put this as common topics as well. It comes up quite a lot. Okay. Coronary heart disease. Okay, so remember we talked about all of those blood vessels in the heart. Yeah, so R, valve, valve, L. We've talked about the aorta, pulmonary vein, and artery, and we talked about the vena cava. Okay, there are other arteries on the heart called the coronary arteries. Okay, these are on the outside of the heart. Remember, the heart is like one big muscle. It pumps, right? If it's a muscle, it needs oxygen in order to pump. It needs energy to pump, right? It gets that from the coronary artery. So whenever you see a diagram of the heart, you never see the coronary artery because that's always like it's always like a cross section of the heart. It's always like a cut version of the heart, right? But on the outside, you have these blood vessels called coronary arteries that supplies blood and oxygen to the heart muscles so that it can pump. Now, these coronary arteries can be blocked. They can get blocked due to fatty deposits. So if you have high cholesterol, you have a lot of fatty foods, it can cause fatty material to build up in this coronary artery, which means you're going to get less blood flow here. You're going to get high blood pressure. You're going to get less blood flow. If you get less blood flow, think about what that means. That means these muscles in the heart are going to get less oxygen. If they get less oxygen, it means they can't respire as much. They can't give the muscle enough energy. The muscle is not going to be able to contract efficiently. And if the muscle in the heart isn't able to contract efficiently, that's basically a heart attack. Okay, so it's very dangerous getting these blocked arteries. So this is what CHD or coronary heart disease leads to. It leads to the coronary arteries, which is the blood vessel supplying oxygen-rich blood to the heart, becoming narrowed by fatty material, and it can lead to a heart attack. 4 things you need to know that can treat coronary heart disease. First one are stents. Okay, stents are these little mesh tubes that you add within the coronary arteries. You open up the mesh and it opens up the artery. So now, instead of having a clogged, narrowed artery, you have one that's opened up. That means more blood can flow through it and all good. Second thing you can do is use statins. Statins are a type of drug that you can take, and they lower your cholesterol. So your cholesterol is the amount of you can think of as the amount of blood, amount of fat in your blood, and it lowers the amount of that cholesterol, which means less fatty material is going to build up. So obviously, advantages of it is that it can lead to less heart disease, so less likelihood of you getting a heart attack or even a stroke. And the main disadvantage of it is that you need to remember to take it every day. It's a drug. It can cause side effects as well. So yeah, you need to be able to compare what's good about it and what's bad about it as well. Artificial hearts. So sometimes people might need a heart transplant. If their heart has failed, they might need a heart transplant. And it's very hard to find a donor for a heart. You have to wait a very long time. So if someone has a heart that's failing, they need to basically keep alive until they find a donor, right? So what they use is some artificial hearts. So artificial hearts are good as a temporary solution, but they're not good for long-term solution. Yeah, because they're artificial, right? The best thing about them is that because they're artificial, they're not made up of anything natural, there's a reduced risk of rejection. They're not going to get rejected by the body. Your body's not going to try and fight it off. It doesn't think it's a foreign object. So that means it's pretty good because the immune system's not going to try and fight it off. Bad thing about it is it's made out of mechanical parts, so they can wear out over time. So eventually you would need to find a donor. The last one is valve replacement therapies. So you can replace valves. Remember, valves are those things that prevent the backflow of blood. You find them mostly in your veins, but you also get them in your heart as well. So like between the atrium and the ventricle, you have a valve. If you have faulty valves, that can make blood flow in the wrong direction again, but you can replace them. So you can replace them by either a biological valve or a mechanical valve. So a biological valve can come from like a mammal, like a cow or a pig, or mechanical valves are made from man-made material as well. So again, the advantages are that mechanical valves, they won't be rejected, whereas ones from another animal can be rejected as well. And anything that involves surgery, if you get a question asking about evaluating anything that's to do with surgery, you can always say there's a risk of infection. Whenever there's a surgery involved, there's always a risk of infection. So it's always like a disadvantage to that. Okay. Okay, cool. Let's try some questions, guys. Let's try some questions. Here we go. So remember, this is going to be the shorter session. I'm going to spend more time on the final topic for session. The human body can defend itself against microorganisms that cause disease, i.e. pathogens. Viruses are one type of pathogen. Name one type of another type of pathogen, basically. Do not refer to viruses. What can we say here, guys? Bacteria. Yes, we can. You can say bacteria. You can say protist. You can say fungus. Yes. 6 marker. Yes, I'm going to do a 6 marker. Definitely. Which two defense systems prevent microorganisms infecting the human body? What do you guys think here? What are the two answers here? So I think you need to think of snazzy here. Stomach. Hydrochloric acid produced by the stomach. Yep. Hairs on the skin trap microorganisms. No, I wouldn't say that. I wouldn't say that. Skin is a barrier. Yes, that's the first S, right? So it's this one and this one. We'll do a 6 marker after this. Yeah, we'll go back to that, the one from the topic 2 one that I wanted to go through, and we'll practice that one. If microorganisms enter the human body, the immune system can destroy microorganisms. How does the immune system destroy microorganisms? Remember, we had APA for this. One of these is relevant here. It's going to be 3. Everyone's saying 3. Yes. White blood cells engulf the microorganisms. Remember, these are the phagocytes. Well done, guys. Phagocytes. Platelets do not kill the microorganisms. Remember, that's for clotting blood. SEAL, SEANL. Red blood cells stick to the microorganisms. No, they don't. Red blood cells carry oxygen, O2. Vaccinations prevent people becoming ill with diseases such as measles. In a vaccine, the measles virus is what? What do they do in a vaccine when they put it in someone? Will the video still be up after you finish the live? Yes. We're going to send you guys an email straight after this whole session finishes at 9:30. As soon as we finish, we're going to send you an email with the recording and the mark scheme to all of these questions, so you guys can do like some last minute revision if you want. We can. So what they do in a vaccination, they put a dead or inactive pathogen into your body. So your body thinks it's a real pathogen. It looks like a real pathogen. It's got those antigens on its surface. So your body tries to fight it off. It releases all these antibodies to try and fight it off, thinking it's a real pathogen and it's going to harm you. So once that's done, you still have lymphocytes there, those memory cells that stay behind, so that if the pathogen was to enter the body again for real, this time, those memory cells can rapidly produce those antibodies and destroy the pathogen before you even get ill. That's the whole point of it. So if the measles virus enters the body after vaccination, the immune system reaction will be fast. Yes, fast. How is the measles virus spread from one person to another? Remember that table, guys? What did I say? I said this one was similar to COVID. So what would you say here? Air droplets. Yeah. Yep. Coughing, sneezing. Air droplets when coughing or sneezing. Okay. Doctors investigated the spread of the disease that causes chickenpox. That's ironic. We were talking about chickenpox earlier. First symptom of chickenpox after exposure to the virus is spots in the body. Cool. 23 children were playing together at a party. On the day of the party, one of the children developed chicken pox spots. Okay. Every 2 days after the party, the doctors recorded when the other 22 children first showed chicken pox spots. Okay. So in day 2, there was none. 4, there was none, none, none. After 10 days after that party, one of those 22 children got chicken pox. Then it increased, then 4, then 2, 0 after 20 days. Okay. In total, 14 of the 22 children got chicken pox. Okay. That's what the question is telling us. Every time you get a table, you need to try and analyze and try and understand what's going on here. What are they getting at here? Because it's a lot of data, right? You need to decipher what they want from you. What was the range of the days on which children first showed chickenpox spots, guys? What do you guys think the range for the days is? (10 to 18). Yes, from day 10 to 18. Day 10 was the first one, and then 18. After that, there was no one, right? Before this, there was no one who got it. So from day 10 to 18. Incubation time is the usual time from exposure to a pathogen until the first symptoms appear. What is the most likely incubation time for chickenpox? So what would you guys think is the most likely time for chickenpox to appear to someone? Yeah, going on to a 6 marker, guys. Don't worry. I would say 14, yes, because that's like the most occurring value here. Most of them, 6 of them got it 14 days after. It's the mode. So I would say it's 6 days. Okay. 2 more, and then we'll go back to that 6 marker. So just one reason why some of the children did not develop chickenpox. So remember, we got 14 children out of the 22 got it. Why did some of them not get it? What do you think you could say there? Some of them could have been vaccinated. Yes, they had it before. Yes, exactly. You can say they're immune. They could be vaccinated. Yes, good. Next one. One mother gave antibiotics to a child who had chickenpox. Why did this child not recover more quickly than the other children? Do you guys know why? A mother gave a child antibiotics for chickenpox. Why would that not work? Sorry, why did I put 6 here? Should be 14. 14. Thanks, Frozen. Yeah, remember we said the most occurring one was 14. I don't know why I put 6 there. Antibiotics don't work on viruses. Exactly. They don't kill viruses because viruses only reproduce inside cells. Antibiotics do not kill viruses. Okay, cool. This question's done. Let's go back to the topic 2 question on heart disease. Here we go. Someone asked for a 6 marker. Here we go. In coronary heart disease, the coronary arteries become narrower. A buildup of fatty material can cause a blockage in the coronary artery. So that's what I explained to you already. Table 2 shows how a blockage in a coronary artery affects blood flow. Okay, so we can see if there's 0% blockage, blood flow is 100 centimeters per minute. It's the highest. But if there's 80% blockage, it's 2. So that kind of makes sense. It says describe the trend. Know the difference, guys. Command word between describe and explain. Describe means literally tell us what is happening in the table. What's happening to the values in the table? Is it increasing? Is it decreasing? As one increases, the other one decreases, blah, 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 blah. If it says explain, you have to say why. Why is it doing that? So what are we going to say? As percentage blockage increases, blood flow decreases. Yes, exactly. That's what you're going to say. So as percentage of coronary artery that is blocked increases, the blood flow decreases. And then it asks us to draw a graph. Okay, so let's try and draw this graph again, guys. Try and do it with me here. We have got blood flow on the right. It's going up to 100. Let's try and use a good scale here. 0 to 100. All right, let's see how many squares we've got. 1, 2, 3, 4, 5. Okay, so I think it'll be safe to make this one 100. Go up in 20s. 20, 40, 60, 80. Okay, that's a good scale to use. Remember, use more than 50% of the graph, otherwise you're going to lose marks. Some people saying they hate graphs. Yes, but it's very likely to come up. So at 0%, we've got 100. Okay, so there's 1. Then we've got 64 at 10. At 10%, was it 10%? Is 64. So we go to 10% and go up to 64. That is around here. Then at 20, we got 42. 20, we got 42 over here. And then at 50, we got 8. That is really low. At 50, we got 8, which is around here. And then at 80, we've got 2. 80, we got 2. Okay. So what do you guys think? Draw a line of best fit. Do we think it's a straight line or do we think it's a curved line here? So like I said, look at the data and try and sense it yourself. Okay. Curved. Yes. It's clearly a curve. You can see it's a curve here, right? If you don't draw a curve, you won't get that mark. There we go. So predict the blood flow in a coronary artery with 35% blockage. All right. So we need to go to 35. This is 30. 35 is here. You'd get a ruler and you go up and then read it off around here. What do we think that is? Some people saying 24, 21. I think on my one is closer to 21, right? 21 on my one. But it depends on how you draw it. They've got like a range. It says 18 to 24 will get you a mark. So it depends on how you draw it. So if you drew it accurately, you should get something between 18 to 24. All right, 6 marker. Explain the effect of a partly blocked coronary artery on the human body. Wow, that is very open-ended for a question. What type of things, guys, do we think we can add here for 6 marks? So what happens if you've got partly blocked artery, coronary artery? So what did I say? Remember what I said. What does a coronary artery do? Decreases blood flow. Yes, I will talk about that. Less oxygen. Yes, that's a good one as well. Health insurance. No, don't need to say that. You could mention high blood pressure as well. Heart attack increases the chance for a heart attack. Yes. Restricts blood flow. Fatty acid buildup. Yeah, because there's less aerobic respiration. Yeah, so remember the difference between aerobic and anaerobic respiration. If you don't have enough oxygen, you're going to go through anaerobic respiration, and that produces lactic acid. We're going to talk about that in the last final session. And yes, okay, let's add some of these points that you guys are saying. So the main thing, if there's a partly blocked coronary artery, it means there is less blood flow to the heart muscles. Because it's talking about the coronary artery, you have to talk about the heart muscles, all right? Then what does that mean? That means there's less oxygen. Again, people have been saying that, oxygen and glucose. Less oxygen and glucose to the heart muscles. What does that mean? That means there's going to be less aerobic respiration. Less aerobic respiration occurs because aerobic respiration needs oxygen. Yeah. If there's less aerobic respiration, it means less energy is going to be produced by those heart muscles. Not produced. I said produced. That's a mistake. Don't say produced for energy. Say released or transferred. Less energy released. You won't get the mark if you say energy is produced. Someone mentioned anaerobic respiration. Yeah, we can say anaerobic respiration occurs, or more anaerobic respiration occurs. Okay. Okay, so now if they have less energy, if these heart muscles have less energy, what does that mean? That means they're going to contract less, right? So the heart muscles will contract less. Okay, so if they contract less, what does that mean? If there's less contraction from your heart muscles, that means blood being pumped around your body is going to be slower. So less blood and we'll say and oxygen pumped around the body. If there's less oxygen, less glucose pumped around the body, that means the person is not going to have much energy, right? So what are the symptoms that that person is going to have? They're going to be tired. So person will experience symptoms, tiredness. We can also say breathlessness. We can say muscle fatigue. Muscle fatigue. We can say, someone said lactic acid buildup as well, I think. Lactic acid buildup. I think that's all enough for 6 marks there. Dizziness, yes, you can say dizziness as well. That's fine as well. Cool. There are different treatments for blockage in the coronary artery. Explain one treatment for a blockage in the coronary artery and how it works. So you can talk about any of those things that we talked about earlier. So stents, you can talk about a heart transplant or an artificial heart as well. You can also talk about statins as well. Any of those is fine. Let's talk about stents. It's 2 marks. We can say stents can be inserted into the coronary artery. And it will open it up. Open it up. Nice. Okay, guys, I told you this was going to be a short one. That is end of part 3. Last final one, probably the most important one, because we're going to go through respiration and photosynthesis at the end. Last 10-minute break, guys, and we'll go through aerobic respiration, anaerobic respiration, and photosynthesis, the biggest topics. All right, wheel spin, no spinning the wheel today, unfortunately. No, sorry. So we will be back at 8:45 for the final session, guys. All right. So I'll see you in 10 minutes. Bye. Hey guys, final session. Well done. You guys have been doing amazing. Last part. We're nearly there. So remember, guys, we're doing the same thing again for chemistry. Chemistry is on Monday, so we're going to have exactly the same setup again on Sunday. So do join if you guys found this useful. We're going to do exactly the same on Sunday as well. So topic we're going to go through now: aerobic respiration, photosynthesis, a bit on metabolism as well. Let's start off with photosynthesis. Okay, so we've discussed it quite a few times already. So photosynthesis is the process where the reactants for photosynthesis are carbon dioxide and water. So a plant takes carbon dioxide from the air, water from the roots, and then converts it into glucose and oxygen, and it uses energy from sunlight. What type of reaction is it, guys? Let me know. What type of reaction is photosynthesis? What would you say? Enzyme practical. Oh, okay. If there are any topics you guys want to go through, remember we've got the recording from the boot camp. So there are a few, yeah, but we have all of those. So if you want to fill in that knowledge gap, you can do that. Endothermic, yes, because it uses energy. Respiration is exothermic. This is endothermic. There's something you learn more in chemistry, but you do need to know it for biology as well. So it occurs in chloroplasts, because in the chloroplast, remember, you've got chlorophyll. Chlorophyll are the green pigments that absorb sunlight. You need to know both the word equation and the symbol equation. Symbol equation, 6CO2 plus 6H2O forms glucose is C6H12O6. You need to remember that. C6H12O6 plus 6O2. If you don't want to balance it, just memorize it. Put a 6 in front of everything apart from glucose. So glucose is C6H12O6. Don't put a 6 in front of it. It has a glucose. Has a 6 in front of it. Okay. If it's laggy, guys, sorry. It does stabilize after a while. Hopefully you guys can hear me as well. How do plants use glucose from photosynthesis? So what I like to use to remember this is SCARF. SCARF. What do the things in SCARF stand for, guys? Let me know in the chat. What do plants use glucose for? Someone said cellulose. Yes, producing cellulose is one of them. So plants will convert some of the glucose into cellulose for cell walls. Respiration for the R. Yes, we've got respiration as well, so that they can transfer energy. A is for amino acids. Yes, so for amino acids, what they do is they combine the glucose with nitrate ions to form amino acids. They join loads of amino acids together to form proteins, and the proteins help the plants to grow. Starch, yes, we've mentioned starch already. That's just like a stored form of glucose. If they don't want any if they don't need the glucose at that moment in time, they can just store it for later as starch. And then the last one is not fermentation, no, fats and oils. They store some of it as fats and oils in seeds. Okay. Imagine you got a 6 marker on this. You guys would get 6 out of 6, right? Okay, let's go back to the cheat sheets. We talked about the actual reaction of photosynthesis. The rate of photosynthesis is also something that comes up quite a lot. There are 3 factors, 3/4 factors that affect photosynthesis. You can use Likert to memorize that. Okay, so this here, the Li, is for light intensity. We got CO. I'm going to put tiny 2 there to remind you that is CO2 concentration, carbon dioxide concentration. And we have T here for temperature. Temperature. All of these things affect how fast photosynthesis is occurring. So not only do you need to know these, you also need to know one more, but I don't put it in this memory trick because it's you don't need to know the graph for it. Does anyone know what the fourth thing is? What's the fourth thing? There we go. Someone said it. Sienna said it. It's chlorophyll, the amount of chlorophyll. The more chlorophyll you have, the faster the rate of photosynthesis. Yeah. It's not a graph that you need to know. So I didn't put it in the list. And that's the one that gets asked the least as well. So yeah, I didn't put it in the list there. So let's talk about those graphs. Before we talk about graphs, we need to understand what a limiting factor is. Can anyone tell me what is a limiting factor? So any three of these or four of these can be a limiting factor. What would you say the definition of a limiting factor is? It is something, you can't say something that limits the reaction. It's something that slows down or something that needs to be increased to increase the rate of photosynthesis. Okay. So any of these can be the limiting factor, and it a factor that needs to be increased to increase the rate of photosynthesis. Okay. So how, like for example, if you had like a warm night, okay, so look at this example. Imagine it's nighttime, but it's summer, it's warm, so the temperature is good, the CO2 concentration is quite high as well. The thing that's limiting it, the thing that's stopping photosynthesis happening any faster is light intensity. Because it's nighttime, the sun's not out, there's not enough light for photosynthesis to occur any faster. On a cold day, let's say at midday, let's say it's a nice sunny day, but it's winter, so it's very cold. So there's a good sunlight, there's good light intensity, there's good CO2 concentration, but the temperature is the limiting factor because it's not warm enough. If it's a warm day at midday, so there's a lot of light intensity and it's warm, then CO2 concentration is limiting. That needs to be increased to make it faster. Okay, so how did these graphs look? Two of them look exactly the same. You need to memorize these graphs. So the rate of photosynthesis increases with light intensity. The more light intensity you have, the higher the rate of photosynthesis. That's because you need light for photosynthesis. Chlorophyll absorb more energy, so it's going to get faster. Now, both of these graphs always eventually level off. Why is that? Why does this one level off, guys? Can anyone tell me? If they ask you for like a 1 marker or a 2 marker, why does this graph plateau? What would you say? Limiting factor, yes, you have to say what is the limiting factor. You would say something is limiting. You have to say which it is. It's either temperature or CO2 concentration, one of the other two factors, because light intensity is very high, right? Light intensity is very high. So you would say CO2 or temperature. You always pick the other two. CO2 or temperature is a limiting factor. Well done, guys. Then you can say the same here. Look at this. CO2 concentration, again, you have more CO2 concentration, the rate of photosynthesis increases up to a certain point, then it levels off. What's the limiting factor this time, guys? What could you say the limiting factor is this time? There we go. Someone said it. Temperature and light intensity. Yes, it's the other two factors. So temperature or light intensity is the limiting factor. That is stopping it from occurring any faster. So those two graphs is easy to memorize because they're exactly the same. Then for temperature, that's the only one that's different. Okay, look at this graph here. What is this graph similar to? What other topic have you seen this graph in? We haven't gone through this today, but you see it in another topic. Which one is it? It's optimum temperature. It is enzymes. Yes, exactly. We see it in the topic enzymes. For an enzyme-controlled reaction, as you increase the temperature, the rate of photosynthesis increases up to a certain point. So you get a maximum around here. That's the optimum temperature. The reason for that is because you've got loads of enzymes flying around. They're colliding with each other. You're getting the enzymes, you're getting the substrates fitting into the active site. So it's happening very quickly. So the faster these particles are moving, the more collisions there are, the faster the rate of reaction. But then it reaches an optimum temperature. This is the temperature at which the rate is the fastest. If you go higher than the optimum, it drops very quickly. What's the reason for this dropping, guys? What's the reason for this dropping? So this one, we can say there's more collisions. That's why it increases. But why does it drop after that? Because enzymes denature. Yes, enzymes denature. Well done, guys. And that's basically what you got to say there. So you could get questions on the rate of photosynthesis. You get questions about farmers trying to maximize their crop yield. So they adjust all of their conditions to make their conditions perfect. So you can talk about that as well. With photosynthesis, you get a required practical, guys, a required practical. So very likely for this to come up. I've seen this required practical come up a lot. It's come up in loads of different versions. Okay, so we do have the ones in, but if you look at like different exam questions, it does look different. They set it up differently. So this one, we're using a gas syringe here. You don't usually need a ruler here, so I'm going to cross that out. But what you have is usually pondweed in water and a light source. You shine light source at the pondweed. The pondweed photosynthesizes and produces oxygen bubbles. Okay, so the bubbles start to get produced. The inaccurate way of doing this experiment, you're basically trying to find the rate of photosynthesis. The inaccurate way of doing it is by counting bubbles. So these Canadian pondweeds are going to produce tiny, tiny bubbles, and they're going to float to the top, and the person is going to count how many bubbles there are. The more bubbles that are being produced in a certain amount of time, the faster the rate of photosynthesis. What's a better way of doing that is to collect the gas. So collect all the bubbles together in a gas syringe. A gas syringe has this scale on it, which basically tells you how much oxygen you've collected. So you can easily read off exactly how much volume you've collected. Okay, and that's quite a usual question. They have in the method, they're counting the oxygen bubbles, but they'll ask, how can you improve this method? You'd say, collect the volume of gas or measure the volume of gas using a gas syringe. All right. And then what will usually happen is you would have to change the distance of the light source. So you start off at 5 centimeters, then 10 centimeters, 50 centimeters, and you have to compare what that does to the amount of oxygen or the rate of photosynthesis. And what you'll find is the further away the lamp is, the slower the rate of photosynthesis, because light intensity decreases. One thing you need to do here, guys, is instead of a normal lamp, you use an LED. Does anyone know why? Why is it better to use an LED than a normal lamp? Why is it better to do an LED than a normal lamp? Monoclonal antibodies, that's a triple only topic. Yeah, so we've got a recording on that. So you can watch the recording. We do go through that. It doesn't heat up as much. Yes, that's something I've seen get asked quite a few times. They ask you, how can you, another way to improve this experiment, instead of using a normal lamp, use an LED because it doesn't heat up. So remember, temperature is one of those factors that affect photosynthesis. So you don't want it to heat up because that's not going to make your experiment fair, right? So you can use an LED instead. Well done. Saying so that it's fair. I saw someone just say that. That's not enough to get the mark. Okay, it's too vague. So you have to say why is it not fair. Okay, respiration. Respiration. The most common topic to come up. And when I say the most common topic, I don't mean there's like going to be a full question on respiration. They always ask it in like parts of questions. So like in a 6-marker, you'd have to mention it. Like even the question that we had on coronary arteries, we mentioned aerobic respiration, right? And anaerobic. You have to always mention this somewhere in a paper. It gets asked. What is respiration? It's the process of breaking down glucose, getting glucose, and breaking it down so that you can release energy. I'm going to say heat energy to the surroundings. So that makes this an exothermic reaction. It's releasing energy. Exothermic. What do we use respiration for? We've got a few reasons. Keeping warm. Our body temperature needs to be maintained at 37.5 degrees. For that, we need a lot of energy. We shouldn't go much below. We shouldn't go much below. Like even going to like 35 is like fatal for us, or going up to like 42 is almost fatal, right? So you need to try and keep that body temperature constant. And for that, you need energy. Contracting muscles for movement. You need energy to move, to cause your heart to beat as well. Carrying out chemical reactions as well. So like building larger molecules. When you're growing, you need to get amino acids. You need to join them together to form proteins in order for you to grow, in order for you to repair your cells or any cuts that you have. That also requires energy from respiration. So those are 3 different uses of respiration. Types of respiration, we've already mentioned aerobic, anaerobic. Aerobic has the word air in it. Air has a lot of oxygen. This is respiration with oxygen. Respiration with oxygen. This occurs in the mitochondria, and it's something that we're always doing. It's the most efficient type of respiration. Equation, very easy to memorize the equation, guys. It's the opposite of photosynthesis. So if you remember the photosynthesis one, you remember the aerobic respiration one, because it's the other way around. Next, symbol equation. C6H12O6 plus 6O2 goes to 6CO2 plus 6H2O. Yeah, so it's just the other way around. And for anaerobic respiration, it's without oxygen. So this happens in low oxygen areas. So basically, when you're exercising, for example, you're not getting enough oxygen into your body. So you need like an extra source. So we carry out anaerobic respiration. But this is not as efficient. It produces less energy. There's 2 equations you need to know for anaerobic respiration. Don't these 2 mix up. Loads of people do. Okay, so if it's a human cell, a muscle cell, it's very simple, the equation. Glucose goes to lactic acid. You don't need to know the simple equation. You just need to know the word equation. If the question's about plants or yeast cells, glucose goes to ethanol plus carbon dioxide. So 2 products there. Okay, so essentially this topic has 3 equations. You've got the word equation for aerobic and the symbol equation. And then you've got the word equation for anaerobic in muscle cells and anaerobic in plants and yeast cells. Okay. Where are these used? So we use anaerobic respiration in fermentation. Fermentation is a process where we get yeast cells. So yeast is like a type of fungus. And we cause them to anaerobically respire. And we use it in bread making because we get dough, we add yeast to it, and the bread rises. So that's why bread has that like spongy texture in it. And also to make alcoholic drinks. So alcoholic drinks have a lot of ethanol in it. So remember, anaerobic respiration produces carbon dioxide and ethanol. So in bread making, the carbon dioxide causes the causes the dough to rise to make the bread. And the ethanol is what makes alcoholic drinks alcohol. It's the ethanol that does that. Okay. Yes, yes, yes. Okay. And then finally, we got a metabolism and exercise. Is the final piece of content that we're going to go through, and then we're going to finish off with some exam questions, and then I'll let you guys go. Metabolism. Definition of metabolism is the sum of all the chemical reactions in an organism. Okay, and they're regulated by enzymes. Examples of metabolic reactions. So there's 5 different ones that are stated in your spec. Okay, let me go through each one of those. Getting loads of glucose molecules and building them together to make starch, cellulose, or glycogen. So in plants, remember they form, remember scarf, they make starch, they make cellulose. That's how they store their glucose. Humans don't use starch and cellulose. We use glycogen. That comes up more in paper 2, but it has come up in paper 1 as well. So glycogen is something that we store in our muscles and in the liver. Then we've got lipids. We make lipids. And we make lipids from, think about what lipase does. It turns lipids into glycerol or fatty acids. We can basically reverse that process. If we need to make fats, we can get glycerol and fatty acids to make lipids if we need to store energy. That's another reaction that goes on in our body. Forming amino acids. I briefly mentioned this when I was talking about plants. To make amino acids, you combine glucose molecules with nitrate ions, and then we can build proteins, right? So that's what helps us to grow. The main one is breaking down glucose to form to respire, so that we can transfer energy, release energy in reactions. And finally, breaking down excess protein. If we have too much protein in our body, we can break it down into urea, and then we get rid of the urea in our urine. All right. Okay, and then the final thing is all about exercise. Doing vigorous exercise, you need a lot of energy, right? So you breathe very heavily, your heart rate goes up in order to provide you with more energy. But the thing is, when you're doing vigorous energy, like your vigorous exercise, like sprinting, for example, you can't get enough oxygen and your heart can't pump fast enough. So you start to respire anaerobically. And when you respire anaerobically, you're basically building up lactic acid in your muscles, which can lead to muscle fatigue. And that increases your chance of getting muscle cramps because of the buildup of the lactic acid, which means all right. So we call this process, we say that the person has oxygen debt. So they basically have borrowed oxygen in order to get this extra energy, in order to do this vigorous exercise, but eventually to repay that oxygen back. So think about you doing like a 100-meter sprint. As soon as the 100-meter sprint is done, do you stop breathing heavily? No, you still breathe heavily for like a good minute after, right? Even though you're not running anymore. And that's because you have oxygen debt. You have all of this lactic acid built up in your body. Your body's trying to get rid of it. So it sends it to the liver, but it needs oxygen to clear that lactic acid. So you still breathe heavily, heavily, heavily to get rid of that lactic acid. And eventually it clears and you're all good. All right, guys, cool. Let's go through some questions just to wrap everything up, guys. So, question on photosynthesis. What are the 2 products of photosynthesis, guys? Let me know. What are the 2 products, not reactants? What are we going to do here? Thank you, thank you. Glucose and oxygen. Yes, yes, yes. Carbon dioxide is a reactant. Water is a reactant. Chlorophyll is what absorbs absorbs light. Yep. So now here we go. Pondweed, experiment, guys. Look at this. We've got the pondweed. We've got an LED light source. So they've already used LED light source. So we don't need to talk about that affecting temperature. We've got the pondweed. We don't have any gas syringes, but we've got bubbles being produced here. So place the pondweed at 5 centimeters from the light source. Measure the rate of photosynthesis by counting the number of bubbles, not the best way. And you're doing it in 30 seconds. Repeat the investigation with pondweed at different distances from the light source. So they're moving it 5, 10, 15, 20 centimeters. How could the student measure of photosynthesis more accurately? I told you what they could do instead of counting bubbles, but I don't think that is that in the list? Oh yeah, it is. It is. So what can we say here for the answers? 3. Count the number of bubbles produced in 1 minute. I think 30 seconds is a very short amount of time, guys. You're not going to see many bubbles there. The longer you're counting it for, the more accurate it's going to be. Yeah. So count the number of bubbles in 1 minute. And measure the volume of gas produced in 30 seconds. Yeah, so measure the volume. Use a gas syringe. That's what they're getting at here. Gas syringe. Okay. The LED light does not get hot. Why is this important? Guys, 1 mark. What can we say here? I'm interested to see how you guys would say. I've told you what it is, but can you guys get the mark? What would you say? Lock and key theory. Remember, guys, if there's any topics that I've missed out, you have that recording. I go through the lock and key theory on that. I go through cell division on that as well. So any missed topics you can watch those recordings. That's what they're there for. Energy efficient? No, we don't say energy efficient. Temperature could affect the rate of photosynthesis. Exactly. Well done, guys. Temperature. You have to say temperature as well. You can't just say it affects the rate of photosynthesis. Temperature affects the rate of photosynthesis. We have the results of the table. Okay, so the further away they're going, that's what it's saying. As the distance increases, the number of bubbles decreases. Calculate the number of bubbles produced in 2 minutes when the light source was 10 centimeters from the pondweed. How do you think? Requires a bit of thinking. Calculate the number of bubbles produced in 2 minutes when the light source was 10 centimeters from the pondweed. What would you do here? It's a mix, guys. It's a mix of questions from all the tiers. Yeah. Times it by 4. Yeah, because this is in 30 seconds, guys. Yeah. So this is 30 seconds. So 2 minutes, 10 centimeters. So for 30 seconds, it was 13 bubbles. So for 2 minutes, it's going to be 13 times 4 because the 30 seconds, 4 times in 2 minutes. So 13 times 4 is 52. So 52 bubbles. Plot a graph. Draw a line of best fit. Guys, I'm pretty sure a graph will come up. You got to practice your graphs, okay? They've been nice. They've given us the scale already. So 5 goes to 40. 5 goes to 40. We've got 10 going to 13. That's there. We got 15 going to 5. 15 going to 5. 20 going to 2. 25, I think it said 1, right? It said 1. 25, 1, and then it goes to 0. Okay. Straight line or curve, guys? Straight line or curve? Curve, curve, curve. Yes, yes, yes. Smooth curve. It should go to 0 because that last point is 0 as well. One conclusion that can be made from the results. What can we say here? We can say as the Distance gets further, the number of bubbles produced decreases, or you can talk about, whoops, sorry guys, or we can talk about as the, 1 second guys, my iPad's just died. Let me get it back up. While I wait for it to come back up, another thing you can say is the further away the distance is from the pondweed, the rate of photosynthesis goes down. Yeah, so you don't just need to talk about the number of bubbles. You can also talk about the rate of photosynthesis as well. So what does that mean? Okay. My iPad's just the app I'm using is updating, so I'm just waiting for it to update. While I'm waiting, let me ask you guys some questions then. First question. Give me 2 products of aerobic respiration. 2 products of aerobic respiration. We're nearly back up, guys. 2 products of aerobic respiration. Aerobic. Carbon dioxide and water. Yes. Carbon dioxide and water. I think we're back up. All right, that was quick. Let me get it back up. Okay, so we'll say the further the lamp is, the slower the rate of photosynthesis. What's even better to say, I'm actually going to say that instead. I'm going to change this up a bit. We'll say the lower the light intensity, because that is what it's getting at, right? The further the lamp is, the lower the light intensity. I'm going to say the lower, the lower the light intensity, the slower the rate of photosynthesis. Okay. How can oxygen production be used to show the rate of photosynthesis? What do you guys think there? How can oxygen be used? Where's the other guy from the thumbnail? That's Nav. That's the other tutor, the other person in the thumbnail. He'll be doing physics paper 1 with you. So I think that's on the 1st of June. 1st of June. Bubbles are oxygen, so more bubbles is more oxygen. Yeah, basically. But you can't just say more bubbles. You have to say more bubbles in a given time, okay? So you have to measure the volume, of oxygen in a given time. You can't just say measure the volume, basically. Okay, scientists investigated the factors affecting the rate of photosynthesis in tomato plants. Here, look at this graph. We've seen these graphs before, right? These are the limiting factor graphs. We've got it at 3 different CO2 concentrations and, different CO2 concentrations and temperatures as well, and different light intensities. We're basically investigating all 3 factors here. Name the factor that is limiting the rate of photosynthesis at point X. What do you guys think? What's limiting the rate of photosynthesis there? Triple content questions. We do have one on monoclonal antibodies. I don't mind doing that, but we can, I can send that to you as well. Light intensity. Yeah, so here it's light intensity. Yeah, this is a light intensity against rate graph. Yeah, if they asked about this state here, so if they asked about this point here, then you would say it's CO2 concentration or temperature. But they're actually asking about the bit that's increasing. So the thing that needs to be increased to cause the rate to increase is light intensity, because they're asking about the increasing part of the graph. If they're asking about the plateau, then yeah, you could say it's temperature or CO2 concentration. But because it's the increasing part, the answer is light intensity. Light intensity. Farmers growing tomatoes commercially the rate of photosynthesis to make maximum profit. A farmer can control the temperature and CO2 concentration in their greenhouse. What is the minimum light intensity a farmer should use to get the maximum rate of photosynthesis? Minimum light intensity. What do you guys think? Look at that graph there. What's the minimum light intensity there? Is it watchable after the live ends? Yes, it is. 20,000. Most of you saying 20,000. Yes, I would say that's right, because look, that's where it goes flat. After that, you're not increasing the rate of photosynthesis anymore, right? So yeah, I would say 20,000 is a good answer. The light intensity you gave may not give the farmer maximum profit. Why not? Why should the farmer not use 20,000 lux? Why could it not give? Why could it not give them maximum profit? This is a tricky one. What do you guys think? Spending? Yeah, it could be. Why are there so many graphs? Biology have so many graphs, unfortunately. Sorry. I've gone through all the papers. Out of the three sciences, I'd say biology is the one with the most graphs. Not as many calculations, but more graphs. Yeah. Costs more. Yes. What costs more? What type of things are going to cost more? There's going to be additional costs for the other limiting factors. Yeah, exactly. Someone said it. So there's going to be more costs for like heating the greenhouse or increasing the carbon dioxide in the greenhouse. So some people do that. They increase these factors to make photosynthesis greater, but it all costs money. So that's going to reduce your profit. So you can say it costs more. It costs more to increase the heating and CO2 concentration. That's actually 2 marks there. In the greenhouse. So we've got 2 marks here already, because we're talking about 2 different factors. And you can say after that, that these additional costs could exceed the profit. Could exceed the profit. All right. Explain the results when the light intensity was 0. Look at this, guys. Explain at light intensity 0. So basically this point in the graph, look at that. What can we say there? What do you guys think? You got to apply your knowledge. So remember, there's 2 types of questions. There's your knowledge questions, but there's application questions as well. Application is where you have to use your own knowledge to try and figure out what's going on here. This graph has gone below the x-axis. What does that mean? That means your value is negative. How can there be a negative rate of oxygen here? Can we not do the question, I hate graph? That means we should do it. Is this a past paper? Yes, it is. It took in oxygen for photosynthesis, but does it take in oxygen for photosynthesis? No. Oxygen is a product. Respiration. Yes, I got some of the correct answers here. When there's no light, okay, remember, this is when light intensity is 0. When there's no light, a plant can't photosynthesize. So it's not going to produce oxygen. What this graph is telling us is it's actually taking in oxygen, because the rate of oxygen has gone negative. Why is that? Reason for that is because the plant needs to respire. All living things respire, right? So if it's not photosynthesizing, it's not releasing oxygen, but it's taking in oxygen for respiration. So it's going, the overall amount of oxygen that it's taking in is more than what's given out from photosynthesis. So that's why it's negative. It's a negative value. So that's quite hard to get full marks there. So you can say there is no light. So no photosynthesis occurs. No photosynthesis occurs. So that means no oxygen is produced. That gets you 2 out of the 4 marks. No oxygen is produced. But we can say respiration occurs. Which takes in oxygen. Which takes in oxygen. So the overall oxygen production is negative. Can you say light is a limiting factor? No, you can't. Because remember, that's at the top, right? That's when the limiting factor happens. Yeah. Cool. How are we doing for time? We'll do one more question, I think. One more question. About exercise. During vigorous exercise, anaerobic respiration occurs in a person's body. 2 effects of anaerobic respiration. So let's go, guys. 4 marks here. So I would say an effect and explain the reason for it. What can we say? Lactic acid buildup. Lactic acid buildup. Yeah, oxygen jet, lactic acid buildup. Yep. We got to explain the effect though. What does lactic acid buildup lead to? Lactic acid buildup leads to muscle fatigue. Yes, someone's written that. Well done, guys. You guys are smashing it. 2 marks. What else have we got? So someone said oxygen debt. Yeah, so if you have oxygen debt, you're going to be breathing fast continuously. So we'll say breathing fast. Due to repaying the oxygen debt. Oxygen debt. Increasing breathing rate is fine. Yep. Increasing heart rate is fine. Yes, you can say that as well. And you can say that's required to break down the lactic acid or repay oxygen debt again. You can say that as well. Well done, well done. We have a 6 marker, I think, next. Design an investigation to show the effect of different types of exercises. Different types of exercises. We'll make this the final one. Final question. Design an investigation to show the effect of different types of exercises on the heart rate of athletes. Again, it's a very open-ended question here. You're not meant to know this. This is application. You need to use your knowledge. What kind of things can we say here, guys? This is not fun. It's not. I agree with you guys. This 6 marker is not fun. What can we say? What can we say? We can say, yep, group of people. Okay, so test a group of athletes. What do you want from them? You need to make sure that they have, based on age, sex, and health. Yes, good. So let's write this down. So test a group of athlete. So these are things that we need to control. And control, we'll just say, and ensure they have the same. So what are some of the things you guys said? Same sex, yes. Same age. Level of activity, yes. So like how fit they are. Yeah, I think that's fine. Then what else have we got? We can then test. It says effect of different types of exercise. So we basically make these athletes. We make them do different types of exercises. So it has to be at least 2 different types of exercises. So we can say 3 different types of exercises. And we make them do those exercises. We measure their heart rate before the exercise. We let them do the exercise, and then we measure their heart rate after. And then we can compare. Right? So we can show how the effect of these different exercises have on their heart rate. So we make for one experiment, we make them do like sprinting for 100 meters. Another experiment, we might tell them to do, I don't know, swimming for 100 meters. And we compare how their heart rate has changed. Okay, so we just need to put that all into an answer. So test a group of athletes. Let's put a big number. Let's put 100 athletes, okay? A lot of athletes. We need the sample size to be big. So I'm going to say test a group of 100 athletes and ensure they have the same sex, age, level of activity, okay? And then let's say take their initial heart rates, then let them do a particular exercise. For a certain amount of time. So you can actually give examples as well. It's like sprinting for 100 meters. Measure their heart rate after the exercise. And remember, we want to make this as accurate as possible. So we want to do this several times, okay? And then calculate a mean. Well, we are doing it several times with loads of athletes, right? So we want to calculate the increase in heart rate. So how much has their heart rate increased by? Find a mean of the heart rates. Find a mean of the heart rates. And then repeat for different exercises. Repeat this for, I don't know, let's say 3 other exercises. And then compare the mean heart rates. And that, I would say, is enough for 6 marks. Well, guys, you guys have been fantastic. Well done, guys. So I've gone through a lot of the questions there. What we're going to do now is you guys the mark schemes. So you guys can, what you do now is up to you. We've given you a lot of resources that you guys can do after. So if there's any topics you guys want to cover again, remember, you can rewatch this live, or you can go and watch the bootcamp recording. So that some of you guys have said a few topics that you want to go through. So you can go through the content on that. You can try some more exam papers as well. That's a really good shout before the exam. We have my entire paper 1 video as well, guys. I've released TikTok like a few days ago. Go and watch that. Like on your way to your exam, watch that video on the bus or something, just to get some last minute revision. Yes, I am leaving, unfortunately. It's been a pleasure, guys. You guys have been amazing. 4 hours. I don't know how many of you have been here since the beginning. But well done. That was a solid revision session, and we're doing it again on Sunday. So I hope to see a lot of you here on Sunday. I hope you guys have found this useful. I gave you my all. I tried to give you as much as I can. Yeah. So all the best of luck, guys. It's your first science exam. Just do the best you can. Don't leave any questions blank, please. Leave them blank. Just say something. Say something. Okay. And yeah, we'll send you guys the mark schemes and the recordings as soon as I get off this live. Okay. I will pray for you guys. Do let me know how you do. Yeah, I'm gonna I'll put a post on like TikTok just asking how everyone's done. I'd love to hear how it went. Tell me what questions came up. Tell me if any of this stuff that we've gone through came up as well. I'll put a post on, add some comments. It's always nice to hear how everyone's found their exams as well. I really hope some of the topics that we went through today come up. All right, guys, we're going to send you guys the emails straight after this. Yeah, so give it like a good 15 minutes after the live, after I go off the live, and we'll send everything to you guys. The mark scheme, the bootcamp recording as well, and this recording as well. Yeah, or you can rewind it right now as well if there's anything you missed. Thank you guys. You guys have been amazing. It's been a lot of people. I think it was like 1,500 people at one point. You guys have stayed to the end. So well done. All the best. Let me know how it goes. Okay. A-level. Yeah, I do teach. We don't do it at FRL, but yeah, stay tuned. We might do it next year. What's the email? help@flashrevisionlab.com. Let me pin it. Let me pin it. Any predictions? No, sorry, I don't do predictions. Anything can come up. All right, guys, have a great evening and all the best. All the best. All the best. You can points for 6 markers. Yes, you can. Yes, you can. How do you get the cheat sheets? You sign up. You can still sign up, by the way. Just go into the link on our bio on TikTok. Sign up and it'll get sent to you straight away. What time are we on Sunday? 5:30 again. 5:30 to 9:30. Exact same setup. All right. I will pray for you guys. Don't you worry. If there's any topic you want me to go through, guys, watch the recording. I have gone through every topic. I've gone through every topic on Bio Paper 1. All right. Thank you, guys. I'm actually going to go now. I'm just taking in all these messages. All right. Good luck. See you. Bye.